And nowadays, the only therapies available are dialysis. So the patient, after reaching a certain threshold of kidney filtration, it has to undergo dialysis. So this is a life-saving therapy, but it can only be applicable for 5, 10 years. So there's gradually a decay of quality of life, and this is normally the patient has to undergo several dialysis sessions a week for 4 hours. So it's a very problematic therapy. And then the ultimate therapy would be donor organ replacement, but due to the lack of compatibility and the number of organs that are available, this is also limited therapy. So it's important not only to understand the disease and how it progresses, but also come up with alternatives in terms of therapies. So that being said, what we want to do in BIRDIE is not immediately coming up with a therapy, but trying to develop in vitro models that will then support a further understanding of kidney disease, also give a bit of support to the development of new therapies, testing drugs, for instance, looking at specific viral induced diseases. But also in the future allow personalized medicine. So we are looking at fitting to inducible potent stem cells, envisioning future applications for patient-specific on-chip screening. So the project is divided in 3 main scientific work packages. The first one is looking at the microphysiological model, and this means that we will look into primary tissue of patients. We will look into the typical models that are used nowadays to be cultured with immortalized cell lines, just as a stepping stone for us to then be able to always compare with the predecessors. So always be able to compare with previous models. So we are looking, and the University of Nantes is leading this work package. They are looking into understanding BK virus infection. So this is a viral infection that occurs post-transplant of organ in a high percentage of patients. And using these 2D perfused modules to understand how viral infection occurs and how to stop it, potential with pharma compounds. Another work package is on kidney organoids. So we are working on establishing iPSC-based cell lines to then be able to work with the bioprinting. This is led by FluiCell. Here they have quite unique bioprinting capabilities, and we want to use these bioprinted constructs with iPSCs. And then in the last work package, we want to incorporate these bioprinted constructs in the organ on chip, envisioning long-term cultures to understand, for instance, nephrotoxicity and the progression of viral infection using iPSCs. So in terms of the first work package, what we see here is just a sample. So we start with the immortalized cell lines to understand, all the parameters that we need to have this model up and running to start viral infection. And we also look at primary biopsy samples to then understand where this viral infection occurs the most, so that we then can eventually correlate the in vitro with what's happening in the patient. So we are looking into spatial transcriptomics to then evaluate where this infection is more pronounced. So in terms of iPSCs, we are establishing protocols. We have tested different protocols to generate different organoids, but also endothelial cells and also epithelial cells. So we are gradually implementing these then with the bioprinting that is the next work package. We are using different bioprinting technologies, but this is our core one. And with this unique bioprinting technology from FluiCell, you can dispense selectively and in very accurate places single cells. So we want to do this then to do the spatial arrangement that we see in the tubulointerstitium. And we want then to combine this with the tubules that we are also bioprinting on the on-chip model. And for that chip model, we are redesigning the chip technology from Tissues, a German company that is also a partner in the project. that has a very unique chip technology, closed circuit that has actuating pumps that allow us to have a continuous flow on different circuits. So we'll have two circuits, one for blood and one for urine-like. And then we want to implement the bioprinted construct on this organ on chip. So there is a lot of re-engineering happening now. This is the off-the-shelf chip that is currently on the T-shirt portfolio, but we are coming up with a new design to implement then the bioprinting. So this is, in a nutshell, what we are aiming to achieve with BIRDIE. As I mentioned, we are at month 30, so we still have 18 months to go. But our ambition is to have humanized kidney in vitro models to allow it to investigate kidney disease, to investigate also long-term nephrotoxicity, testing different drugs and compounds. Also to have an in vitro platform that allows multiple screening and testing, and hopefully achieving a more complex in vitro model for viral infection and nephrotoxicity, and also be able to correlate this to primary tissue of patient and to the disease progression. So that being said, I would like to acknowledge all the project partners and all my group back in Maastricht. If you want to look a bit more into the project details, you are welcome to join our project page or also our Twitter link. So if you have any questions, I would wrap up here. Thank you for your attention. Thank you.
Yes, so thank you, Carlos. We have quite a bit of time for questions, so we can perhaps start with questions from the audience here at Louisville. So the floor is open. Maria?
Hi, nice to meet you and nice to hear about the work. I wanted to ask, what do you think is the main challenges of integrating bioprinting issues, the basic model that they have?
So the main challenge is size and scale. So if you think about bioprinting, despite that their technology allows single cell resolution, if you think of having then this integrated on the chip, then you have to think of multiple components. And if you think about the tubulointerstitium, there's a lot of cells in there. So mimicking this in a very confined space that then you are able to perfuse, that's the challenge that we are taking gradually. But having this, put it all together and having the similar function that you have on the tubulointerstitium is something that we will hopefully be able to demonstrate in the end, but the size is the main challenge. So the convergence of sizes will be probably the first challenge to overcome. But I think that will be feasible. We have already some results showing that. But then putting a very tiny fraction of this model into the chip and being able to culture it for several weeks is something that we still need to demonstrate.
So are you taking the approach of either shrinking the bioprinting or expanding the BIRDIE platform? Which one are you taking?
We are taking the approach of having something plug and play. So we don't want to have shrinkage and contraction. We want to have as stable as it can be. But there's certainly, if we think about a soft tissue, there will be always remodeling happening and so on if you culture it for long periods of time. So we are working quite a lot on hydrogels and matrices that we can control swelling and their degradation. Also to ensure that there is a bit of stability throughout the culturing period.
Okay, so we stay just in the room. I'll run you the else.
Yes, perfect. Thank you.
Good morning. Thank you very much for inviting me and Phil to speak today. We're going to be talking a little bit about the microphysiological systems that we use at AstraZeneca and in renal bioscience. And also, we're really happy to join in on the discussion on what's needed in the future. So I'm Emelie Lassén. I'm an associate principal scientist at AstraZeneca in the bioscience renal team.
And I'm Philip Dalsbecker. I'm a postdoc in CVR and safety science under clinical pharmacology and safety sciences, also at AstraZeneca. Try again, it should have broken up. Yeah.
Maybe just need to press first. And then we'll start and
Yes.
All right, so what we'll go through is first the mission of renal bioscience, then a little bit about the considerations that we have when it comes to finding the right model for the proximal tubule. Phil will go through a little bit about the MPS systems that are currently in use at AstraZeneca, and also we have a bit of an MPS wish list, and also a future outlook. So if we're starting with the mission of renal bioscience, in 2011, AstraZeneca did a major revision of its R&D strategy in order to improve productivity and success of new projects. And this new strategy relied on what's called the 5 R's. So it's the right target, right tissue, right safety, right patients, and the right commercial potential. And within renal bioscience, we mainly work with the 2 first R's. So the right target and the right tissue. But of course, right safety also comes in very early at a project's inception. And we work very closely with clinical pharmacology and safety sciences, or CPSS, which is where Phil has one of his affiliations. And we can see the use of MPSs, or microphysiological systems, as well as NAMs, or new approach methodologies, in all these 3 R's, where the use of human material in an MPS has the potential to improve the link between the target and the disease through a translatability advantage. And this, as we know, some diseases such as diabetic kidney disease is quite challenging to model in rodents, for instance. And also the translatability potential of MPSs is also there when we're looking at the right tissue to assess, for example, pharmacokinetics, pharmacodynamics. And this kind of segues also into the safety R, where we want to see the uptake and transcellular transports in the proximal tubule. Let me see. Here, just a second.
Skip one slide.
Oh, I skipped one slide. Oh, I'm so sorry. So, in alignment with this 5R framework, the strategy for identifying new drug targets has changed to be more focused on human target validation, where targets with a link to genetic evidence of disease and knowledge of its function as a disease-driving mechanism has been found to be associated with less risk of failing in clinical trials due to lack of efficacy. And so how we work today is that materials such as serum, urine, and biopsies from CKD patients and healthy individuals are analyzed through high-throughput assays such as proteomics, transcriptomics, metabolomics. And this information is then processed by bioinformatics, and lately more and more AI and machine learning is involved in this process. And this generates a list of possible targets. This is then assessed in silico, and we're looking at what has been known in previous studies. Is there a translatability to animal models that we can use to model the disease, species homology, and so on? And also by our subject matter experts, or SMEs, in the lab and in the team. And this narrows down the list of drug target candidates. And from this list, we go on to in vitro target validation. And this is, of course, where the MPS systems come into play, as well as more traditional 2D systems. The targets that then passes these tests are going on to be analyzed in vivo. We have, everything from zebrafish, rodents, non-human primate models. And through proof of mechanism and proof of principle in CKD models, we can then select the best targets that goes on to an investment decision on whether or not they can enter the portfolio and be entered into clinical trials. So if we take a closer look at the in vitro target validation, where the team I'm a part of do most of our work, we have options of varying complexity at our disposal. So we still work a bit with cell lines and 2D cultures quite extensively. And we work with mostly human cells, so primary iPSCs, pluripotent stem cells, and sometimes also with immortalized cell lines. And the next level of complexity that is also often used in projects are transwell cultures, where we're interested, of course, in cell-cell interaction. And we also have the MPS systems where we will go into a bit more detail. And then in increasing complexity and laboriousness, it also increases the physiological relevance. We also have the organoids that we also do work with. And on the top here, we have ex vivo tissue, which preserves the physiological complexity of the organ, not always the physiological functions. And we know that, for instance, precision cut kidney slices are difficult to keep in culture for a long time. And as you will hear, I think, throughout the day, quite a bit, and also from us, there is always a trade-off with these systems. And when we're looking at the bigger picture, we see that an increase in physiological complexity comes at an expense of the screening capacity or the throughput. And going back down the table by scaling back on physiological complexity and achieving a higher throughput, we're also risking increasing the pitfalls and having a too reductionist approach. Mm-hmm. So what are we specifically looking at when we want to find the right model for the proximal tubule? So one important consideration, of course, is cell polarization. We know that the cells do have apical and basolateral poles in vivo, and these poles, they have different receptor expression, transporter expression. This polarization is lost in 2D culture, but it can be retained in more advanced culture systems. The group that Philip is a part of has published some interesting articles where they have compared the polar drug and solute expression in 2D cultures in transwells and MPSs to the actual kidney cortex, and found that, of course, the MPS and the transwells, they do improve the expression of these transporters, but we're still quite far from the expression in the human kidney cortex. And one such transporter that we, of course, are interested in is the SGLT2, the sodium glucose cotransporter, because that is involved in the standard of care in, for instance, diabetic kidney disease. And what we're also interested in is translatable protein expression. So, and this will depend on the cell source, where we're working on a cell line or primary cells. If we're working with pluripotent stem cells, there's also a possibility of incorporating organoid-associated cells. And I've also seen publications where they do co-cultures of cells from different origins, though still with the same cell type. And as I mentioned, SGLT2 expression is something that we're really interested in looking at for glucose reabsorption, as well as megalin for protein reabsorption systems. And of course, from the safety perspective, OATS1, OCT2, and MATE1 for synabiotic excretion. And we know that the protein expression will also depend on the cell's culture surface, so the ECM will also be of great importance here. We also know that fluid flow will allow for continuous nutrient access and waste removal, and that shear stress supports the differentiation of the PTETs. There have also been publications that show that there is a steep increase in the transepithelial electrical resistance with fluid flow, and also a better formation of the tight junctions. So we're getting in. But I'll leave over to Phil to describe some of the platforms that we use.
So that being said, let me take in order to give you a brief look into the systems that we have in some of our work streams, as well as the, as people mentioned many times, important trade-off that comes in versus in simpler systems versus more complex systems, and user friendliness versus physiological relevance. On the more on the more user-friendly side of the spectrum, we find platforms like Unit organoplates. It has the same footprint as a standard 384 well plate, which makes it more easily integratable into standard work streams. The platform is gravity-driven, foregoing the need for pumps and tubing in favor of a simple plate rocker. And the chip consists of several interconnected wells, as you can see here, where several parallel channels converge at a point in the middle where they are separated only by phase guides, as you can see in illustration K. The central channel is filled with collagen, which is cross-linked, and cells can then be seated into one or both of the adjacent channels, where they will then polarize based on the presence of the ECM and the flow. And the cells can then be both sampled and dosed either apically or basolaterally, as necessary in the experiments. The fact that you can also fit up to 60 of these chips in a single plate means that it's much higher throughput and more amenable to multiplexing compared to the other models that I will cover today. On the flip side, though, the gravity-driven flow is bidirectional and is not constant over time, even in a single given direction, meaning that the shear stress will also be variable over time. And not all of the cells are actually grown in contact with the collagen gel, meaning that only the subset of cells that actually grow against the collagen can be dosed and analyzed basolaterally. All the same, it is a solid platform and has a higher throughput potential than most other kidney NGS systems that we have access to. On the other end of the spectrum, we find platforms like the Nortis Parviva. This one is a pressure-driven platform where a single pump feeds up to 12 chips in parallel using unidirectional flow, making it more physiologically relevant than the corresponding culture in the organoplates. I'm focusing here on the dual-channel chip. There are a few different variants, but this is the one that we primarily use in our work streams. It centers on two separate fluidic circuits that overlap or at least cross close to each other in the central area here, which is a matrix film chamber through which two parallel hollow tubes run. The cells are seated into one of the tubes, and where they will adhere and polarize based on the presence of the ECM as well as the flow. And the cell tube can then be dosed either apically from within that tube or basolaterally by applying the compound or ligand of interest to the other tube and having it diffuse through the collagen and reach the outside of the cell tube. The shear stress is at or near the physiological level as cited in literature and can be adjusted within range using the pressure-driven pump. However, this platform is much lower throughput than the organoplate, as a single perfusion rate here fits up to 12 chips in parallel, each chip effectively being one data point. Meaning that if you want to multiplex, you need an additional perfusion rate, whereas if you want to multiplex with your organoplate, you just stack several plates onto the same plate rocker. All the same, this is a very powerful platform with high degree of physiological relevance, and we have used it successfully for publications relating both to nephrotoxicity as well as the uptake of pharmaceuticals. Of a similar degree of complexity is the tissue's humic line of chips, where these chips are very interesting in that they're built to be essentially multilineage or even multi-organ co-culture compatible. They can support steroids, organoids, and 2D cultures, as well as in some cases transwell inserts, making it a very flexible and versatile platform. The fact that the on-chip pump also allows for medium recirculation means that it is very useful for organ-to-organ feedback loops, as well as to study drug metabolism over time, which is harder to do in a single pass perfusion system such as the Nortis platform. The one that I have a picture of here also has, as was mentioned before, two separate but connected fluidic circuits where they have an overlap region in the form of a membrane, as seen here, where a culture can then be dosed apically and basolaterally or simply be exposed to different types of medium as per the requirements, though this is done using a 2D membrane culture rather than a 3D tube, as in the case of the Nortis. Similarly to the Nortis, this also has a lower level of throughput compared to something like the organoplate or simpler cultures, but it makes up for that in the increased complexity that you can get out of it. With the tissues chip, we have both worked with it ourselves in-house as well as collaborated directly with tissues to essentially take advantage of the fact that the people developing the chips have more practical experience with working with the chips, whereas AstraZeneca can then contribute with compounds and data analysis and such. In most cases, or in all cases, we try to find solutions that are best suited for all parties involved. We have also worked through an external collaboration with the 3D-based PP system. This is a fairly similar system to the Nortis in that you have two parallel tubes running through an ECM gel. So here the gel is hydrogel-based rather than being collagen, as in the Nortis case. It runs using a peristaltic pump, and the footprint is that of a standard 6-well plate, where each such plate setup fits up to 6 pairs of channels. It allows for diffusion and exchange of soluble factors, either if you have one empty channel and you can apply actively and basolaterally, or as in the case with the publication that came out of this, having two different cell types in the different tubes and having them communicate by exchange of soluble factors through diffusive transport. This was done through external collaboration with an academic collaborator, where they produced the chip and used it and did the cultures in-house in their labs, and AstraZeneca then carried out the bioinformatics analysis of the data that the solid product. Similar to both the tissues platform and the Nortis one, this gives us a high degree of physiological relevance, but at the cost of higher complexity in practical handling and lower throughput. And this is a recurring trend that we see with these chips. So we have this trade-off between the physiological relevance and the ease of use. On the one hand, you have the Melis organoplate, where you have high ease of use and quite a good throughput for what it is, but at the cost of some of the physiological relevance. Nortis tissues and 3D vascular all cover for this in their own different ways, bringing back the physiological relevance to a higher level, but at the cost of a reduced ease of use and reduced throughput. This also extends to transwells, which forego the flow entirely in favor of even higher ease of use. And organoids have their own innate 3D structure, but that cannot necessarily be separately accessed and dosed the way you can do with the chips. So I think at this point it's becoming clear that the choice and design of an MPS is no single task, and that there always is this trade-off to work around. The question becomes, what do we and what does the pharmaceutical industry in general need? And here I hand over back then.
So far, that will depend on the research question. So we do go to more complex systems because they do have better physiological mimicry and higher structural complexity. We see that here in the illustration from some of my former colleagues, Dr. Ananda Krishnan and Dr. Asendu. For instance, here the vascularized organoids in a perfusible microfluid organ on a chip that also was described. And for this to become even more physiologically relevant, we would like to integrate immune cells. That is very relevant to a lot of our projects, as well as integrate some kind of non-invasive biosensing that would be very valuable to be able to monitor kinetic parameters, glucose, lactates, oxygen, and tear in real time. And then, as we mentioned, the simpler systems do offer higher flexibility, throughput, and they're easier to implement and use within our existing work stream. So the question is, of course, could these be combined? Could there be an optimal assay? So far, of course, that is a big challenge. And another challenge there is the standardization, which I think has been mentioned in this consortium before. And we need to consider confounding factors, such as technical confounding factors. It could be operator, where the machine is placed in an incubator, things like this. And those have been published for an organ on a chip that is quite successful at AstraZeneca, where we're actually working with bone marrow and things like that. We should hear about that. But if we're looking at the proposal of a unifying sort of chip that would fulfill all of our needs, I think Phil will give us a little bit more detail about what that would entail.
Yes. So given all of this, what factors actually matter the most? I would argue three things. First of all, assay compatibility. Because different biological questions will require wildly different actual readouts. In some cases, this might be morphological changes or immunofluorescent staining or uptake of fluorescent ligands, all of which are then dependent on having compatibility with imaging modalities at sufficient resolution and magnification. Chips like the 3D vascular and the Nortis that also depend on a thicker layer of ECM can also put a strain on the working distance of objectives and contribute further background to fluorescent staining. In cases where you have chips where the footprints actually match that of a well plate, even then it can be difficult in some cases to integrate that with standard analysis instruments, such as plate readers for absorbance and fluorimetric assays, if the location of the cells relative to the footprint does not match those of standard well plates. Some assays may also require you to extract the cells from the chip, which may be challenging in cases where either the dissociated cells can disperse into the fluidic architecture and be difficult to retrieve, or where you have to flow dissociation reagents through the chip slowly enough that it impacts the cells in the meanwhile. And in some cases, you might simply be required to extract the cells from the chip just because you can't carry out the assays that you want in situ on the chip as you would in a well plate. And in cases where your assays, for example, depend on retaining the polarization of the cells, that resolution would then be lost if you extract the cells from the chip. Basically, the utility of a chip hinges on its compatibility with the different assays that we need, which in turn can vary quite a bit. And that brings us to the second point, flexibility. Because the more biological questions you can use a chip to take on, the more use it will see, both across functions, such as bioscience versus safety sciences, and even across organs. This also means that it requires a higher degree of advanced specialized user training, that the payout for that is still spread across several projects or even several functions, making it likelier that the chip will be implemented into our standard work streams. And the utility of these chips in different work streams also brings us to the third point, multiplexing and upscaling. Because MPS work is a material art, there is no way around that. Many chips depend on external peripherals such as pumps and tubing and effluent reservoirs and other specialized things required to either work with or assay the chips. And while on a smaller scale, it's easy to fill a small lab with all the equipment you need to carry out these assays for a smaller proof of concept, much of what we do is done at a higher scale, and the scale of all the peripherals must not then become too cumbersome and prohibitively costly as it is scaled up. Essentially, if we want to make the most out of a given chip, we need to make sure not only that many projects will want to use it, but also that they are able to use it on the scale that they need. And altogether, this is quite the tall order. As a former MPS developer myself, I am acutely aware of that fact. I think the best we can do from the side of innovation is to work with the existing needs and find the compromises that can be made, both from the sides of chip developers and us as chip users. Work around this, find the compromises we need, and do what we can to find the optimal solution for everybody.
Yeah, so I'll just wrap it up with a quick sort of future outlook. So what we want to do is remove the obstacles for a broader use of proximal tubular MPSs within AstraZeneca, because there is a remaining need for complex in vitro systems of the proximal tubule and also of their peritubular vasculature, both for mode of action, efficacy, and safety research. We want to be able to discern transcellular transport, reabsorption, and excretion in increasingly physiologically relevant systems. And though numerous systems exist, such as the ones they described, they all have different advantages and drawbacks, and they are also not all benchmarked to the same level. And we need a system that balances fidelity against the utility and the scalability, as Phil just described. But we think with concerted effort, and that's why we're very happy to be here, we can find good or better compromises to further leverage MPSs and support the pharmaceutical industry in the long term.
And with that, we thank you for your time.
Thank you for an excellent presentation. Again, we have some time left over for questions. So do we have any from the BIRDIE team present here on site?
Yeah, maybe I have one to start with. So you mentioned that you select these MPS systems based on the research question, but do you also compare between MPS systems for the same research question, or is that too expensive to do? Because multiple have drawbacks that are the other's advantage and so on, but are you extracting the exact same data from these multiple microphysiological systems if you have the same research question?
To a limited extent, I would say. Now, I've only been with the company for a year and a half, so I don't know too much in depth, but the impression that I got is that in some cases, we have tried to do smaller benchmarking studies of different chips against one another, but overall, it has more been finding the chip that is right for the research question at the time, and then simply using that chip for that research question and only comparing it against like our normal benchmarking controls.
Okay, so and you picture this different phases, so the target discovery and then gradually 2D, 3D, and so on. So do you foresee eventually a full replacement of the 2D and 3D and then having that MPS broader, or it's going to be so expensive that you will still have ad eternum 2D and then gradually going to 3D and gradually going to MPS and then in vivo? So do you see this, this schematic changing over time, or you find it really cumbersome to put all these things and then replace all the others? So is it the bottleneck of maybe regulators? Is it the bottleneck of cost? Why there is so much inertia of having always the same kind of steps?
That is a very good question. I don't know if I have a good answer to it. Essentially, because so much is built on what has been tested and was like the tried and true throughout history, essentially, that any major change will still need to be benchmarked against what was the leading standard before, right? But also with how many different types of chips there are, even just like that small variations in how a chip is built and structured can have important impact on the resulting phenotype of the cells cultured, and you might not be able to control for all of those factors. If you take two chips of comparable complexity in both structure and handling, if they are built differently, they will affect the cells to different extents and have different advantages and disadvantages in physiological relevance, even relative to one another. Because the more you specialize a device, the more you can go from, for example, just if you compare an organoid culture in a static condition, for example, there you have multiple cell types represented and their interactions, but not in an external physiologically relevant environment. Compare that to the Nortis chip, where we have only proximal tubular epithelial cells, but we give them a more relevant environment. If you want to study something that is specific only for those cells, you need a chip like that. But then you lose out on a lot of the like the intra-organ interactions of the other cell types. The more you specialize a chip, the more you gain in one thing and lose out in others. Meaning that I think it'll be hard to, as an early step, replace too much of what is like broadly applicably well enough done in conventional systems. But eventually, I hope we'll get to a point where a lot of that can be replaced with sufficiently complex, but at the same time sufficiently manageably user-friendly chips. It's just that we're probably looking at a further horizon there.
Yeah.
And I think, if I can add to that, I think, of course, the regulatory aspect is going to be important. So whether or not it's going to be accepted to use MPSs for safety data, for instance, I know the FDA has changed some of their rules quite recently. So that's also an aspect that's going to be important. But I have to say that we're a little bit challenged with using MPS systems at AstraZeneca. So the change is coming quite slowly, and I think it's because of the challenges that Phil just described. So we do still talk a lot about, we still use a lot of animal models, we still have a lot of 2D models within our research. So, but there is an ongoing effort within the whole company to do new approaches and new approach methodologies. So we're really hoping, and then of course it's in alignment also with the 3 Rs that we're trying to replace animal studies. We're also, reduce the number of animals, but it is a big challenge. And I think it's a challenge of communication too. It's a very large company, and knowing what they're doing in safety to what we're doing in bioscience, that's another challenge that I wasn't anticipating as much when I started.
Yeah, there's a lot of stakeholders involved in the process. But and a follow-up question, since we touched briefly on regulators. So do regulators still ask the traditional 2D cultures in all these steps, or they just want a particular step of this pipeline, that you demonstrate efficacy? Do you have to focus more on in vivo, or they are looking nowadays more only to the in vitro and MPS? Do you see a change, or is it still the historical, traditional regulatory requests?
I think that's an excellent question. I'm also quite new to the company, so I've only been there 6 months. So I actually don't have a good answer there. I've just heard about sort of the changes that are happening in the US, so I'd be very happy also to hear the Medical Products Agency and what's being done in Sweden, because we're also very interested in that. And I'm more in the bioscience part and feels more on safety. So I think safety, of course, that question is more acute.
Yeah.
So I unfortunately don't.
Okay. That's a good picture too.
Yeah, it was very interesting.
So we have a question here from Zoom, from Alexandra. Hi, very nice presentation. Do you work with patient-derived cells or just the cell lines?
So we work mostly with, we do work with patient-derived primary cells. And actually, most of them are from... So they are not from disease patients usually. So that's another thing that we want to implement. And I think Carlos also touched upon that. So the iPSCs to have more disease relevant systems. I know that's really an interest in both safety and bioscience. So there is some work with patient derived cells, but it's not more healthy.
Thank you. We have, two more questions. Frank.
Not a really naive question, but we all agree here to say that there are great expectations in the addition of immune cells in these kind of models. So, will you use, common words, the bone marrow derived cells, or are there protocols to generate iPSC derived immune cells? What is the focus for you? Mononuclear phagocytes, other cell types?
Oh, I don't know how much you can get into that. We actually, I think, first of all, like finding a system where we can incorporate immune cells. I'm not exactly sure if I can talk about which kind of immune cells we want to work with. And that also I'm not. We have some immunology experts in the team, but unfortunately I'm not one of them. So I'm sorry.
Yes, Gavin.
So I'm sure you can sit here on the different systems that you're using. I'm largely a technology-based person, so my question will be technology-based. So as we, let's say, transition, as you should, from more basic cultures and all the way to the advanced systems, you're essentially more and more confining your thing you're trying to measure inside something that you can't physically interact with. The more advanced your MPS system, the harder it is to touch it. So you're screening everything. And I know that for many years, a lot of the pharmaceutical industry leveraged very heavily on patch clamp systems, electrochemical readout systems, all of which require you to touch your object. And in this, you obviously can't touch things because they're encased. And you equally explained this issue of, if you release the cells, you're actually destroying most of what you actually did by putting them in there in the first place. So what sort of, you said that, yes, there's a big need for non-invasive methods to actually how to study things in place. But are there things which you're thinking of more specifically in our comparison? Is it more alternatives to patch clamp, for example, or is it alternatives to something specific which is of greater impact than something else?
A lot comes down to what kind of assays you want to run on a chip. And if you have a chip that is like designed dedicated for a certain type of process, then I think you can build the structure as much as you can around, okay, what needs to be doable onto this chip. If you want something that is more generally applicable, that becomes that much harder. And then you have to think about basically how can you have the structure you need in a practically usable way, but still essentially just kept in place with a clamp holder or screws or whatever, so that you can separate it to the extent that you can interact with the cells as needed. And in cases where, for example, if you have an intact tubule, then you can't just open it from the top and add something. How can you build around that to at least make sure that the handling is as simple as possible? it's unfortunately going to come down a lot to the specifics of if you're trying to build a chip for a specific process, a specific environment, or if you want something that is more general. But in either case, a lot is going to come down to how you can design that around all the variable needs that you might face. And the more I think you think of that as the chip is being designed, and the more you can interact with the intended end users, the better. Because I remember from my time designing MPS systems during my PhD, you spend all that time focusing on how you should best mimic the physiological structure of the organ. And then it comes as an afterthought that, oh, but you can't do most of the assays that you plan to do. It's the kind of thing where it's one thing if you discover that for yourself, but if you develop a product and then sell it and it turns out that the end users can't really do what they wanted with it, that's a different situation.
And I think we will need systems where we can have multiple readouts, certainly.
Yes.
And yeah, I think, for instance, to be able to monitor glucose or oxygen, that's something that's always going to be relevant. I mean, we do work with the big disease drivers like diabetes, hypertension, hypoxia. We do want to model that for sure.
So we have one final question from Zoom here. From Carlin, great presentation. Have you tried to use primary urine or media with adapted pH on your models?
Not urine, to the best of my knowledge. I remember hearing something about one of my colleagues having used pH adapted medium in one case. I don't remember what came out of that. But it should also be said that since the different MPS systems that I've covered today are part of different work streams, I only have experience with one of them, and I haven't had that much time to interact with the people using the other chips. So I can't really answer that for sure relative to the other chips.
Yeah, like I said, unfortunately, there is more to wish for when it comes to the application of MPSs, and that's what there is a company-wide efforts to implement them into more projects, but that's also a big thing that we need first to associate them with a question that's relevant for the project. So just doing like exploratory work is unfortunately not something that we can do a lot of. So it has to be project-based.
Okay, just one.
So very quick question.
It's more common than because. So thank you very much for this very, very impressive presentation. It's really good to see that someone comparing all the different models and getting the feedback is highly, highly valuable. Also for me being a MPS developer, and I think AstraZeneca is really at the forefront of testing, showing what we can do with the systems. So just one final statement, maybe. You've tested so many different models also and explained very well that they could also be used at different stages of the drug development process. Now thinking about what is then actually feasible in the long run, how many assets would you say are you capable of having in your labs in the long term, in the long run? Would it be two systems, one being high throughput and one being high complexity, or is it really then looking at the different fit for purposes, or what do you think would be feasible in the long run of having in a lab? How many chips systems?
That's difficult to predict, I would say, because at the moment, I feel like the MPS utilization within AstraZeneca is still in a growing phase. It's difficult, I would say, to predict at this point in what direction it's going to grow. Because on the one hand, the more you can standardize the system and have that as a general part of many work streams, the less additional peripherals and the less extra space would need to be dedicated to that. But at the same time, it will vary a lot based on how high throughput is required for a given stage and phase, and how much organ-relevant fidelity has to be recaptured. And if it's fine to focus on specific parts of the kidney, like the proximal tubule, for example, or if you need more complete models. So it's unfortunately really hard to answer that because we can't yet know to what extent that AstraZeneca will expand upon this potential that we already see in the use of MPS in this space.
I can also add to that, but there is a success story, I think, which is more a bone marrow MPS, and I think there it's also really what's really important and differentiating it from previous work is that we're using additional, we can use maybe AI, machine learning things to make them more applicable and maybe more relevant when it comes to clinical predictions. So I think that's also something that we're looking into quite extensively, and I think that that's going to help also as MPS systems become more useful going forward.
Okay, thank you so much for a very impressive presentation and a very good discussion. We will take a short break now for coffee, and I think we will return in 11:15, so in 15 minutes.
First of all, thank you for the invitation for your BIRDIE workshop. I have been following your community for a while, and that's a privilege for me to be here and talk to you a little bit about what we are doing at Utrecht University. And just a little bit of background. This is us. We are at the Utrecht Institute of Pharmaceutical Sciences. There are 5 divisions, and you can see them all here, very diverse. But at the same time, they all have a common thread. We share the building, we share the space. And at this institute, there is a division of pharmacology where I am situated, and I'm performing my research. And at this division, we are looking at all sorts of disorders that can be pharmacologically triggered or manipulated via drugs, biologicals, or for instance, food components. And that is because we have a very strong collaboration with Danone, with Nutricia, where we also can look at how food components can act as medication, for instance. So here are just a few of the research that we are performing, the topics of research that we are covering. And at the Division of Pharmacology, there is a group called Experimental Pharmacology. And this is here. This is us. And other group of experimental pharmacology, which is led by Professor Ros Macereu. We are also focusing on the kidneys, because this is also what we are here. We also have a lot of kidney research happening, and mainly what we do, and already Carlos already presented about the kidneys and what they are doing. We also look at what the kidneys are doing and trying to understand what happens when the kidneys are failing, so that we can use this information to reproduce in the lab, in the idea of finding a new mechanism of actions also for potential therapeutics. So I will not bother you with going too in-depth in this information. We all already know what the kidneys are doing and why the nephrons are very important. But what I would like to highlight is that the nephron space is a very busy space. There's a lot of activity happening in the nephron, all sorts of functions, all sorts of cells that need to synchronize their activity in order to keep everything properly functioning. But whenever the kidney is not functioning properly, we discuss about chronic kidney disease that can lead to end-stage kidney disease, where patients go on dialysis. And we know that dialysis is considered a sort of an artificial kidney. And that is because, dialysis removes small to middle size molecules, replicating thus the glomerular filtration of the kidney. But one of the important molecules that we are also very interested in are these protein-bound uremic toxins, or PBATs, which are, as their name says, they are very highly bound to albumin. Thus, they cannot be eliminated via the classic dialysis that only mimics the glomerular filtration. And this is because of this binding. So they accumulate in the plasma of the CKD patients, and they are associated with a wide range of comorbidities. And ideally, what we would like to do or to replicate also in dialysis is to the decoupling of these toxins from the albumin in such a way that the albumin will go back into the patient, so into the plasma, while the free fraction of these PBATs will be eliminated into the waste. So it will be eliminated during dialysis. And what we did, like in many instances, when we want to replicate certain functions of the body, we look at how the body does it. Because we know that the kidneys can efficiently eliminate these compounds. So that is because of the action of the proximal tubule cells, which possess active transport properties, in which they remove these compounds from the blood into the urine. And it's not only these compounds, but also other drugs that are heavily bound to albumin. So what we do in our lab to understand how we can eliminate these kind of compounds, we use a cell line, which I will talk a little bit in the next slide, and that is conditionally immortalized proximal tubule epithelial cells. And we have manipulated this cell line in such a way that it expresses OT1. So here I'm showing OT1, but we also have an OT3 counterpart, so organic anionic transporter. And this transporter is responsible for the handling of this kind of compounds. So these endogenous compounds that are very difficult to remove via classic dialysis. There are also other transporters that are present in this cell line, also efflux transporters, efflux pumps, and we use this cell line for disease modeling and drug screening, and I will show you a little bit later in a few slides. And we also know that the OT1 expression is stable along passages, and we are harvesting these properties of the cells to do this disease modeling and drug screening. So you see here also a few of the stainings that are that we perform for this cell line. And now that we have the cell lines and we have the proximal tubule cells, we look again at why do kidneys fails? Why do they fail? And for that, we use the cell line that I just mentioned to you to understand what happens when kidneys are failing. So recently, one of our PhD that is now submitted his thesis, he looked at disease modeling, and one of the models that he wanted to replicate in vitro was renal ischemia reperfusion. So the IRI injury, which normally is performed in animals. So we wanted to know, wanted to see if we can use these cell lines and this type of injury, and we can replicate this injury in vitro. So for that, João, what he did, he cultured the cells, he exposed them to an ischemic injury. We mimic this in vitro by using antimycin and 2-deoxyglucose to deprive the cells from their metabolism and for their respiration. And we also used hypoxia for that. And then we look what happens to the cells. So more focused on the ischemia part, coupled also with hypoxia, so atop of the chemically induced injury. And we notice that the cells are suffering. They shrink, they start to detach from the surface of the well. And we also looked at how they behave metabolically. So we performed some metabolic activity assays, but also we looked at the ATP production at the mitochondrial mass. And we noticed that in ischemia, especially the one that we have coupled with hypoxia, the ATP production and consequently also the mitochondrial mass is heavily reduced. We also did a sort of metabolic screen to see what kind of metabolites or pathways are being modified in this kind of condition. And here you only see the normoxia in a hypoxic condition. And we noticed that, for instance, in the normoxia, there is an enrichment of some metabolic pathways that are related to energy production and protection against cellular damage. And while we add the hypoxic injury as well, so atop the chemically induced, we also reduce the oxygen concentration in the culture. We also notice again metabolic pathways that are enriched more for the energy production, but also the production of essential biomolecules that we believe are for the cells to compensate for the injury. What we have done further, we looked at the treatment option for this kind of injury. So now that we have the cells injured, we wanted to see, can we rescue the cells? And for that, we used MSCs-derived conditioned media. It has already been mentioned of using MSCs as a therapy. And in this case, we used the products generated by the MSCs during culture. We had either the conditioned media, either extracellular vesicles, and here we split them in large extracellular vesicles or small EVs. But I will not go too deep into this information that we have obtained, but rather just give you a brief overview. So here is after the treatment with the conditioned media in this case. And what you see in A, B, and C, and U is basically MSCs that were obtained from different sources. So adipose tissue, bone marrow, and umbilical cord. The upper part is with the cells that were in normoxia, and on the bottom, the hypoxia. So you can see that using conditioned media from MSCs, regardless of the source, we can already start to see that the cells are recovering. They start to cover again the surface of the well plates in this case. And also we have noticed that the ATP production is increased. And as well, we looked at some redox homeostasis compounds that are also related to cell bioenergetics are also increased upon treatment. So this paper has, this work has recently been accepted. It is a mammoth of work. So there is a lot of information there. So I will invite you to have a look to see more in depth what we have performed. Another disease modeling that we have been looking at is the diabetic nephropathy, and I will explain you why we call it 2.0. And in this case, we were not able to use our cell line because our cell lines don't have glucose transporters. There is also a research route that we would like to pursue, in which we also modified our cell line that has the OT1 expression and efflux pumps to also be decorated with glucose transporters. So that is work in progress. But for now, I will show you what João did. So instead of using our cell line, he used HK2 cells, which are known to possess this transporter, the glucose transporters. And usually researchers are using high glucose to model the diabetic environment. What we have done, we have added to this environment, to the high glucose environment, palmitic acid and glycated albumin, which are also compounds that are accumulated in the plasma in diabetic conditions. So we basically made the model more severe by adding these compounds. And then we also looked at treatment formulation, and in this case it was a SGLT2 inhibitor, EMPA, and then we looked at basically how this compound reduces the kidney injury markers, and the injury markers that we had looked at are KIM1 and NGAL. So here you can see how the cells look like upon exposure to high glucose for 24 hours and 48 hours. But also with palmitic acid, so PA, and glycated albumin as well. What we have looked at is again at mitochondria and what is exactly happening, the turnover of mitochondria and this ratio between fission and fusion. And we notice that the palmitic acid does have more effect, but in the mitophagy part, and also in the mitochondrial fission compartment. But at the end of the day, both the addition of palmitic acid and glycated albumin slightly increased the injury profile. And I mentioned to you that we looked at how the SGLT2 inhibitor would rescue the cells by looking at known kidney injury markers. And in this case, we noticed that the drug didn't do anything for KIM1, so this injury marker is still present. Even after we apply the drug. But NGAL, we noticed that there is a decrease in the production of this marker upon treatment. So this is again a work in progress. We hope that soon we will have more information on this study as well. But again, one of the, as I mentioned, a core business of our work is to understand why do kidneys fail and how we can reproduce these conditions in the body, in vitro. And we also look, for instance, at medication. And because we have this cell line that helps us screen for certain drugs, we can also look at medication load. And for that, we look at this kind of polypharmacy. And one of the very interesting things is that patients that do have some sort of kidney disease or kidney function impairment do take a lot of medication. And especially those patients that are on end-stage kidney disease that still have some residual functions, renal residual function, they take a lot of medication. So you can imagine that many of the medication that is being taken by this patient is potentially handled by this OT1 transporter. So in many instances, the transporter can be overbooked by handling all sorts of drugs, but at the same time also eliminating the toxins that should be eliminated. So for that, what we did, we used our cell line, and then we did a study in which we used, for instance, indoxyl sulfate, which is a very well-known protein-bound uremic toxin, in combination with several drugs that are being prescribed for kidney patients. In this case, we used fluorescein as a surrogate compound, also substrate for OT1, and we looked at the percentage reduction in the uptake of this fluorescent compound. And we noticed that, for instance, with the addition of the drug or these drugs that I'm mentioning here, so valsartan, losartan, pravastatin, and simvastatin, they are reducing the uptake of fluorescein, suggesting that they are also handled by the OT1. And then when we add indoxyl sulfate on top of this in culture, we notice a further decrease, suggesting that again the fluorescein uptake is further impaired. The issue here is that because we are using a fluorescent substrate, we don't know if it's what is the intracellular load of the drugs or of the protein-bound uremic toxin, so in this case, indoxyl sulfate. But we have recently been awarded with a grant, which I'm going to talk to you here, in which we are interested in looking at drug-toxin interaction that might lead to the accumulation in blood and could change, for instance, how we should medicate patients so that we don't so that we can avoid overdosing or underdosing. And this project is now funded by the Dutch Kidney Foundation, in which we are not going to use fluorescein anymore. We have developed LCMS tools to detect protein-bound uremic toxin and some drugs, so that we can have a clear profile of what kind of loads of these specific compounds we can find intracellularly. As you can imagine, you can do all sorts of combination also in this project that we are going to start now with the University of Maastricht. And this combination allow for also this in silico modeling. So the colleagues in Maastricht, namely Aurélie Carlier, has developed some methods in which we can use some machine learning algorithms to detect, for instance, what will be the right combination of drugs and toxins so that we can still eliminate both compounds. And of course, we also have some other projects ongoing that are looking at this kind of in silico and AI models to enhance risk assessment of chemicals, for instance, all sorts of chemicals in kidney cells, but also to assess the safety of other pharmaceuticals without using laboratory animals. And like this, we can generate this kind of prediction graphs, curves that we can further take into studies. And the idea is to eliminate as much as possible the use of animals. And here I will just give you an example of the work that we had with Aurelie. You see here that there was also a collaboration with them. So the cells are sitting in a well. We look at the OT1 activity. They have designed this model, this box in which you can see the OT1 transporters picking up compounds, indoxyl sulfate in this case. And one very important addition to this model was the addition of albumin, because we know that indoxyl sulfate is also highly bound to albumin. But in a uremic model, in a uremic conditions, we also know that albumin is modified. So that is also a very important feature of a uremic or of a disease model that we need to add in order to understand the activity of the transporter and the affinity of the compounds towards albumin and towards the transporter. So these are ongoing work. If you want to know more, I put already a reference from this study. But just bear in mind that here we are only looking at the OT1 mediated uptake, and we don't address the transepithelial transport. So the removal, the complete removal of the compounds on the apical side. So for that, what we do and why we need to understand this kind of transport is because we are also working with the bioartificial kidney. And in the bioartificial kidney, the idea is indeed to remove these compounds, protein-bound uremic toxins, in such a way that albumin goes back to the patient and the toxins are free and easy to be removed. And for that, we use this system. We cultured our cells on hollow fibers, and I will show you a little bit more about that. So we culture the cells on hollow fiber, our CIPTEC OT1 cells. They need to create a very tight barrier, because the membrane that we are using is has a pore size that allows proteins to pass, so albumin to pass. So the barrier has to be really tight so that we don't lose important molecules. But at the same time, the cells should be able to execute these transport functions, so the removal of compounds from the inner tubes or from the blood compartment into the apical space, which will correspond to the dialysis fluid compartment. We have done some studies also with perfusion in this kind of tubes decorated with cells, in which we look at different compounds. So here you see quinurenic acid, hippuric acid, indoxyl sulfate. We can measure this via LCMS, and then we perfuse with uremic plasma, so plasma from CKD or end-stage kidney disease patient, and then we created these transepithelial clearance profiles. Here is how it looks like. These are cells sitting on the fiber. Again, here just a few characteristics. I would just like you to look a little bit at the fiber itself. So we do a coating with collagen type 4 to enhance the monolayer formation. And then we can also perfuse inside and out. And inside we can also perfuse with drugs, for instance, and then we can see what comes out on the apical side, which will be basically the urine compartment. So this is a picture, a SEM image of the fibers that we are using, and a little bit of the procedure that we are doing. So we do a first coating with L-DOPA, then with collagen type 4 or with other type of proteins, so laminin or blending or other compounds, ACM compounds. Then we do the cell seeding. And in principle, our cells, we allow them to proliferate on the fiber. And then we can also, there is a step of maturation where they start to organize and behave like epithelial cells. And then we can do all sorts of characterization. And mainly we are interested at the removal of compounds from the basolateral to the apical side. So here I will just show you a few of the examples using these tubes, these fibers. So here was one of our PhDs looking at, for instance, healthy cells, healthy CIPTECs, but also diseased CIPTECs. And this is, we call them cystinotic CIPTECs, which are the representative of the disease, cystinosis, that has a high manifestation in the kidney function. So here is where she cultured these diseased CIPTECs also on the hollow fiber, and they are pretty leaky because they cannot form a barrier. Another project that we are currently working is the formation of a glomerular filtration barrier. And for that, we used different coatings. We used conditionally immortalized podocytes in this case, and we coat the fiber with different coatings. And you can see here collagen, laminin, or a combination. And of course, in the combination, we noticed that there is much more adherence of the cells. We also looked at specific markers of these cells. Immature cypods mean cells that are in their proliferation phase, and then mature conditionally immortalized podocytes are after they were put at 37 degrees where they should mature. We noticed that the cells change also their shape. We believe that they just instead of being spread like a pancake, they just lift their bodies. So that is characteristics of the podocytes. And the idea is that we would like to see them bridging towards each other and covering the spaces in between them, so that they can create this filtration barrier. And here you can still see that there's still some work to do for the cells, but we are confident that if we leave them a little bit longer, they will create this barrier. We also looked at specific markers that are associated with the activity of these cells, and nephrin and podosins are also there, so that is also very important for us to see. Using the same idea of perfusion, we perfuse these cells, these fibers containing the cells with compounds, and in this case it was with phytinolene, which is a very small molecule, and the idea of seeing how the cells allow certain compounds of different sizes to pass. And here you can see that in comparison with the empty cells or with the empty fiber where there are no cells, with cells there is a full leakage of this very small molecule. But when we put phytidextran of 40 kilodalton, then compared to the empty fiber, the cell seems to block the passage of this compound. Still, we believe that we can do better if we allow the cells to grow a little bit more, so that they can cover all the holes or all the areas of the surface of the hollow fiber, so that they can really create a filtration barrier. So I mentioned to you that we work a lot with hollow fibers, perfusible system. This is a system that we have in-house that we can either rock or we can do dual perfusion. And the idea is that you can perfuse also on the inside of the fiber, but also on the outside of the fiber. Another project that we are doing in our lab, it's related to this part, so the proximal tubule and the interaction with the endothelial cells. So in the idea of looking at how molecules pass from the blood into the urine via this interstitial space. And for that, in fact, a team of 2 of our PhDs looked at creating the coaxial bioprinting of the proximal tubule and the vessel. And probably you have already heard of coaxial bioprinting. But in this case, we have made use of alginate combined with gelatin. And then we had this system created in which the extrusion was performed. And then we can generate this kind of ribbon-like structure. And I will show you a little bit more in detail in a few slides. And the idea is once we generate these hollow structures, like ribbon-like hollow structures, we can seed cells, allow the cells to create a monolayer and then to mature. And then we can generate, for instance, you can see here, for instance, at day 0, the empty tube, and then seed it with cells. And then at day 7, the empty tube, we wanted to show here that the structures remains intact. And also with the cells, you can see that they created a very nice monolayer. And we also looked at the possibility of doing 2 tubes, 2 ribbon-like tubes within this large fiber. And then also we seed it over there. The most challenging part is very difficult to access each tube individually to perform perfusion. So because these are very small fibers, that is more challenging in terms of technicalities. But once we have the one single one, what we can do, once we have the cells, we can perfuse again with phytinolene, and we can see, for instance, again in this model that we have used of cystinosis, cystinotic cells, how leaky they become once we perfuse with inulin. So at the moment, we are looking at the different parameters that we can adjust with this coaxial printing. For instance, the tube diameter, the frequency of the coiling, or the amplitude. So basically the thickness of the entire system. That are also projects in the making. And just a few last slides of what we do in terms of tubes. And I guess you have already got the idea that we do like tubes. We do work with a lot of tubes. We also work with metal electrowriting, and this is a printing technique in which a filament of polymer is a very thin filament of polymer is deposited on a surface at a very controlled manner. So we know exactly where the filament is going to fall on the surface of in this case, the surface of a mandrel. And then we can also evaluate how the filaments fall in comparison to each other. So this kind of winding angles can be created. And what Emelie did, our PhD now postdoc in our lab, she seeded endothelial cells and proximal tubular cells on the surface of these tubes, and she noticed that cells respond differently when they are seated on different winding angle tubes, and we noticed that the proximal tubules, for instance, they like the rhombus shaped or the crosshatch, and if you allow them to grow, they can bridge the polymer, so there is a big pore, and then they can fill it in. Basically, the polymer is only offering a scaffolding. It's like a scaffold, while the entire tube is covered with cells, a monolayer of cells. And here you can see, again, we did a perfusion with inulin, and I hope it's moving, where you can see that indeed the tubes and then the way the cells close the pores doesn't allow inulin to pass through. And now just a very quick, because I noticed that I'm out of time. One aspect that we are also very interesting, and we are using also perfusion for this, is engineering the vasculature, and this was a collaboration with I3S in Portugal under a twinning program called Remodel. And that allowed us for colleagues from I3S to come to our group to see what we are doing, and then go back and mature the idea. And here is the final result. The idea is that we can use these fibers also to create these vascular channels, because we can culture cells on top of them. And then the idea is that from this tubes that we create, sprouts will come through. And the idea of vascularizing organoids in this case. And here you will see, I hope, let me check. You can see my pointer. You can see these units over here, which are called the vascular units. And I will go to the next. And this is where the vascular units have been developed. These are the ones developed in the group of Cristina Barrias at I3S. Where they do this kind of spheroids of endothelial cells and fibroblasts, that they embed them in fibrin, and then as you embed them, they start to sprout, and then they reach to each other, and they even create these anastomosis sites. So we wanted to leverage the potential of these kind of vascular units. So what we have done when they came to us, we seeded the cells on a tube of ours. We embedded this tube into fibrin, and we saw that the cells are starting to sprout. This was an initial study. Now we can see much more endothelial cells in the sprouts. And one of the studies that experiments that have been done is to perfuse with dextran, and then to see where dextran ends up. And then it went basically where we also had an expression of endothelial markers. And the following idea is to also perfuse with beads and see where exactly they end up in the matrix. And then we noticed that the beads were localized where the small sprouts from the vessel were also present. And here is the final approach that we had. We used, in this case, intestinal organoids close to a tube that is sprouting. And then the idea is to use these vascular units as an intermediate structure to allow perfusion of the organoids, so basically to reach to a vascularization of organoids and using the large tube as a source of blood vessel-like structure. And at the moment, we are expanding this for the kidney vascular unit. So we are using for this kidney fibroblasts, and they are sprouting as well. So now we are in the phase of putting them together with kidney organoids and see if the kidney vascular units will surround the kidney organoids and create vascular structure towards the organoids. So with that, this workshop is also about in vitro models and how we can replace animals using these in vitro models. So they are very complex, and already the speakers before me have already mentioned this complexity and all these combinations of materials, cells, manufacturing techniques. We are at the pharmaceutical sciences group, so we are also looking, as already hinted in my presentation, at drug-drug interaction and comorbidities and the setting of comorbidities in our models in order for us to better understand injury. But also we are very interested in the part of nutrition and how food can enhance the repair of injury. And with that, well, I would like to thank, obviously, all the colleagues that have been involved in this work, and also the funding. And then if you have any questions, I will be happy to take them. Yes.
Thank you very much, Silvia, for a very impressive presentation. I think we have room for one or two questions.
Oh, I don't hear you.
Hello, can you hear me? Can you hear me now? Yeah?
Yes.
Okay, sorry. Thank you so much for a very impressive presentation. I think we have room for one or two questions.
Maybe I have one.
Yes,
So,
Carlos.
On the dialysis tubules, is there a particular reason why you have the cell layer on the outer part rather than on the inside?
Yes. We are using commercially available fibers. These fibers have a diameter of 300 microns, and we cannot see through the fiber. So we decided to put the cells on top of the fiber because it's very easily to seed and to see if the cells attached. We agree totally because it's the inverted polarization, basically. We are now having, for instance, with the tubes, the metal electrowriting tubes that I showed to you, that gives us more versatility and control in the design of the fiber in such a way that would allow us to seed cells from the inside. So that is also something that we are very interested in. I didn't have the time to show it here, but we are looking, for instance, at how curved surfaces like concave and convex surfaces of different diameters can enhance polarization and then function. So we are looking also at function follows form aspect, but with these porous membranes, the ones that are also used in the clinics, we don't have that versatility, but we are looking at in parallel at this. There was just not time to talk about it here.
Yeah, thanks.
Okay, thank you very much, Silvia. I think
if there's any question, always in the chat. I can respond also in the chat.
Yes. So we have a question here. I think we can have it if you type the answer in the chat here.
Yes.
Thank you. So now we will move on to the next speaker, which is Maria Tenje from professor at Uppsala University.
Yeah. They arrived just a bit late, so I didn't have time to say hello to you guys. Who are you? So, thank you very much for the invitation to join you here today. I want to talk about microfabricated hydrogel-based barrier weapon. It's very similar, actually, to what we just heard from the previous presentation, but we'll take a different approach. I think it's going to touch upon a lot of the different topics that we already discussed. But I think it's very nice also because now I know this, we have a mixture of like tech people and bio people here in the room. So I think it's very exciting to be here. So just as Jonas said in his introduction, I'm professor at Uppsala University and I'm especially in microsystems technology. So I'm interested in the tech approach, but of course in the life science applications. So this is a slide that I wanted to show you because I just think it's so nice. I got it from a colleague at KI, Julia, where they have described very, carefully all of the different steps that you need to combine and integrate to develop this organ-on-chip model for the different applications. And we already discussed, the sort of mismatch between complexity or, biological information that you can gain and user practice. But all these different steps that we have here, we are interested in the engineering sides of fabrication, looking at different materials, also integrating some sensors, and then also the biomaterials, but actually how can we fabricate devices or how can we tune the properties of the biomaterials that we engineer. So 2 examples that I don't want to say too much about, but I still want to show you because maybe it could spark some ideas that we can discuss later over lunch, is that, for example, we can control the adhesion on different hydrogels simply by patterning adhesion molecules. So here we have used hyaluronic acid, which is an ECM component, but that doesn't support cell adhesion and proliferation. So that's why you have the black bracket, so we have no cells. And then we pattern lines with RGB peptides. And here in this study, we wanted to look at how do endothelial cells react to going on the line and how do they behave if the lines are different widths. So going from 10 up to 100 micrometers, how do they spread? How do they extend? What happened to the cytoskeleton? What happened to the nucleus? And one thing that we observed, because we also looked under high and low glucose conditions, and how do the cells and the cells respond to that? And one thing that we found is that we have this aspect of cell chirality, that they orient themselves in the bone and monolayer, and these cells have a negative bias, so they always sort of orient themselves downwards in these images, and that's just very interesting. Because how the hell do they know that this is outside and this is inside? They are just seeing a line. So that is something that we're sort of exploring a bit further, just from a basic scientific viewpoint. But these are 2D cultures. We also work with controlling cell positioning in 3D. And this work is done on droplets. So basically hydrogel droplets, that's the red blobs that you see. And here it's actually fluorescent particles, just for visualization. So we can use acoustic forces to integrate into these platforms where we produce the droplets, and the radiation force from the ultrasound will then position itself at the center. Here you see red because we have red nanoparticles also inside the gel. This is egg gels. And the cells or the particles will then move to the center. And then we expose this UV cross-linkable gel that we're working with, so we can freeze the cells and particles into this position. We have also done with cells, but these images are much easier to sort of understand the concept. We don't see any compromised cell viability. They proliferate and grow to the same extent as if not exposed to the ultrasound and if not exposed to the UV light. But of course, one would want to really dig a bit deeper also into what really happens with cells exposed to ultrasound specifically, and that's very interesting. So this is work that is extended in a near consolidated grant that started earlier this year. But it's also not something that I'm going to drive into, but just to bring up some take acts into. So the topics was biological barriers, and that's also I don't just do kidney. We don't do much kidney at all, actually. There's broader, of barriers, because that's interesting from an engineering viewpoint to have something that's separating two compartments. How can you control what you have in between? And many of these tech solutions, it becomes an organ when you put the cells in there. So depending on what cells you put, you obviously get sort of different organ models. But actually, we started by looking at, blood-brain barrier, and now looking at general vessel models. Because that's where I think we can contribute the best with the methods development that we do. So how are in vitro barrier models done today? Is after representatives mentioned the transwells. That's you have the porous membrane, you culture 2 cells, you put something onto the top and you look what's going through to the bottom, and then you can do off-chip analysis for this. 2D cell cultures, static environments, synthetic membranes, not good. How can we address this? So, because we know a cell growing on a flat surface, that's not the same as a cell growing in a So we started to say, okay, how can we sort of control 3D cell culture environments? Because, the benefit of this is that, and that's something I think it was very nice that I think it was Philip who highlighted that in the Metas platform, for example, you have your gel in the center and the cells are growing in contact with the gel. You could also have cells inside that gel, for example. But they are exposed to different environments, the ones that grow onto the cell and the ones that grow in the channel parenchyma. The nice thing here is that they all grow on the membrane. So that's why we thought, okay, how can we actually, control these 3D matrices in some ways, and how can we integrate them with a microfluidic platform? So how can we control this? So we started working with collagen, because collagen is a nice material so basically we can prepare a suspension of collagen, bring it to neutral pH, and then add the cells, and then it will gellify 7 degrees. And then we can, by actually just placing a paper on top, we can pull out the liquid so that we can compress the collagen, and it becomes more manipulable substrate. So this image that you have here on the right is cross-sectional confocal scan where you can see the collagen matrix and you can see a cell sitting inside it. And we can then grow on collagen. Is of course the most abundant ECM component, so growing cells on top, they proliferate, they grow very nicely. Looking here at an Almer blue assay, you can grow inside the matrix as well. Also cells proliferating nicely. But then the question came, so how can we integrate this? Because now here we wanted to address the flow aspect. Because one of the main works that we do is based on microfluidics. So how can we integrate this into a microfluidic platform? Well, we can place it obviously at the bottom of a channel, and we can grow cells in there. That works really well. And then you don't have to barrier in the house. Perfused culture, but that's not what we want to have. So if we want to put it into a microfluidic system, we actually had to put it between sort of some supporting membranes, and then we could perfuse top and bottom. And that was sort of also kind of working, but then we were facing the same or a similar problem that was presented just before, because if we have these now so that we have mechanical support from the membranes and we're perfusing these channels, well, then the cells are not really exposed to the flow. And also we rely a lot on diffusion into the matrix. So it's like, no, it works, but that's not good enough. That's not what we want to do. So then Hannah worked as a postdoc with me, and then she said, well, can't we just replace the transmembrane with a biomimetic membrane so that it actually becomes free? And that's what she then worked on.
with the goal to still use the transfer format, because that's also, sticking with something that is already known, is of course, a benefit of instead of starting from scratch. So how can we then integrate the biologically derived materials into this and make sort of a hybrid structure for mechanical support? So this is a classical transwell. This is a transwell with Hanna's biomimetic method. So the first thing is to get the mechanical support. And this is always the challenge now when you work with biological matrices. You want to have them. And I mean, it's the same challenge we're all facing. You want to have them soft enough so the cells can grow, but you don't want them to collapse. So what do you do? So we integrated sort of a mesh, a PCL network that was prepared using a 3D printed mold with like pins on the side and basically just weaving with this network. The thread's 100 micrometer in diameter. So going up and down and then across, and then putting these between two metal plates, copper, and heating up and melting so that this formed this support structure, an internal skeleton, you can think of it as. So this is a zoom in of the pins where she was weaving. And then she also added some solid supports around for manipulation, for gluing it onto the trans base. So that what you see in the center here is the open pores. Using different spacings of the posts when she was weaving. She could go from 2 millimeters just below, because these are also 100 micrometers, you have to remember. So just below 2 millimeters and down to about 0.4. So openings of these areas. And then she integrated different types of gels because we want to get away from the synthetic materials. So PEG is also, of course, a synthetic polymer, but it's nice because you can have materials. Yes. So Hannah used PEG. She did her PhD on PEG, so she was very fond of PEG and how it functionalizes in all sorts of different ways. So we said that's where we'll start. So, for example, integrating some methacrylate to get more biomimetic. Using 4 arms, so integrating also with the 2 arms to tune sort of gel properties. So now the basic concept is that you make your support network, place it between 2 glass slides with the gel peptide in between, put it down into contact. These will then form the thickness of the spacing, so you end up with 100 micrometers. Cross-link that under UV light, and then she could take out her gels, hybrid structures, and then glue it onto the trans. A known format with a biomimetic membrane instead of synthetic or the PETs or the PC ones that you would normally have. So what do these structures then look like? Well, they bend a bit. And that's not surprising. I mean, that was also something that was brought up at the beginning, the swelling issue. So this is now a cross-section, and you can see where you have the pores and where you have the support structure. You have a pore, you have a support structure. And that's why she then looked at different gel compositions, because, I mean, it's a matter of swelling, having a restricted movement when you're in these pores. So basically, the material has to go one way or the other. It cannot go sideways, because there we have the material. So here we see we have basically no swelling. And that's when she worked with hyaluronic acid. And that's why I thought it was interesting to show you that even if hyaluronic acid doesn't support cell adhesion, you could then think of functionalizing it so that you can still use it as a material here. But she also looked at different PEG compositions, and you see here quite large degree of swelling. So here it is defined as bending angle and depth. So basically how deep and what's the angle of bending. And then also she looked at gel, which is very often used also as a 3D culture material. Then again, the bending, you could also think of that as the feature. So, for example, she could also use this actually as a platform for spheroid formation in a very controlled form. And then especially with the hyaluronic acid ones, that you could also make bend with other compositions. The cells would then form a spheroid and grow in a very soft environment. So that's also one aspect that we're exploring a bit further, how we can use it for that. But she could grow. So this is what you see now. One opening here, MDCK cells to form a barrier. And then you could see that even if the gels are bending slightly, the cells will of course conform to that, and they will form a tight barrier using sort of this copper electrode endobe to measure here. You could place her transfer system inside, and then see as the cells were proliferating and forming a barrier, the tear values were increasing to a sort of similar level as if they were grown and transferred. Another thing that she also explored was to use this method to actually prepare stacked multilayers, where you could then imagine having different types of gels that are suitable for different types of cells, so that you could form a more complex structure. And here, this is visualized by integrating also fluorescent. But you see, this is where we have the support layers of the net, and then she has these different materials. So summarizing this work, it is possible to realize free-forming gel structures with this approach. The bending is a challenge. It can also be used as a feature. It can be tuned by the gel. But it's possible to combine multiple layers. And that's a very attractive approach that we are now continuing also. But still, we didn't actually have flow added here because we have the transwell format, and then you have your wells. But that's also another approach, that we are exploring also a bit. How can we integrate flow there? So moving on to a different approach that we have taken on addressing the flow and still keeping in mind that we want to work with biomimetic hydrogels. So if we say again, okay, we want to form a vessel, now we want to focus more on how can we integrate flow and how can we perfuse this, and we want to have a hydrogel-based structure. You could think, okay, so start by making a microfluidic device then. This is a cross-section, it's 3 glass layers that are bonded together. And in these areas, we have connection ports so that we can flow that material through. And then you can fill this up with a UV cross-linkable gel and define a channel inside. Remove the non-exposed gel, and then introduce liquid so that it becomes perfusible. And then you can seed cells inside your vessel. But you can also, because now we have this confined space here, we can then also load a gel which supports then 3D cultures of cells in the gel outside. And this is also along the lines of having, the best material for the cells that you want to culture. So here, if we want to work with endothelial cells, gelma, for example, is a very suitable material. But then we can surround it with fibrin for fibroblasts. And this also then connects with the previous question I asked. So this is Federico Cantoni, who's worked on this approach. And he's actually defending his PhD on Friday. And it will be broadcasted. So go and I'm happy to share the link with you if you want to know more about the details, what he worked on. for this 5 years or so, because I'm going to summarize very briefly all the work. But it connects with that question that I asked before. If you want to integrate something into a microfluidic channel, you need to think of the dimensions. So actually what he worked with is that he worked with 2-photon polymerization. So instead using the femtosecond lasers to write and define the structures, because then we can really go down on the dimensions. So the good thing with 2-photon is a free-form fabrication. It's a high-resolution 3D printer, basically. Laser-based, it requires a photo-activated initiator. I mean, you need to be able to cross-link the material in something. But it's, compared to the UV, which we also work a lot with, where you expose the whole bulk, with 2-photon, you just expose and cross-link at the boxon. So that means that you can make very well-defined structures. So, for example, this is one of the studies that Federico did, was that he compared the performance of 3 commonly used 2-photon printers from UpNano, NanoScribe, and FemtoLab, and looked at sort of, printing resolution, what can you do, looking at smoothness of surfaces depending on objectives that you use, and also looking at complexity of printing both large structures combined with smaller structures. And this is all with hard resins, so not with hydrogels, but it's more to benchmark the different systems. And we have the Nano 1 system in our lab from that's the one that we're. And you can see, for example, in this here, the FemtoLab, if you just look on the top row images, the FemtoLab is the one that comes out with the sort of best performance of these structures. But the printing time is also 4 minutes, whereas you have 11 seconds from the Nano 1. So it depends, of course, always what you're really after, what is important for your system. But we work with the UpNano, but we want to work with hydrogels. So not these classical hard resins that you would otherwise get. So, and 2-photon printing of hydrogels, I mean, it's been done before. You can operate it either in a cross-linking mode, where basically you have your gel and then you go in and write and you define where you want to have your structures. You can have cells growing in this, and you can see then that they follow the scaffold design here. If you start by having a gel and then you start writing your structure, you are also exposing the cells inside this gel to your printing, which will basically kill them. So that's not super good. What you can also do is you can actually use 2-photon to ablate, so basically remove material. So think instead that you fill a chamber with your gel, and then you go in and you write where you want to have your channel defined, and then you can perfuse that and you can seed it with cells. Here you have the same, so you can form these very nice tubules. You see here, this is 50 micrometer diameter, so it's down to the 50 micrometer scales. Here you have the same issue. If you have gels inside here, you ablate, you will burn, the cells that accidentally were in the way, which might not be an issue, but it could definitely be. Also here, we only have one type of material. And again, coming back to the aspect that different cells might actually prefer different types of environments, it would be beneficial if we can combine different types of gels. So some optimization was, of course, first done in this study. And we worked with this bio-inks, it's called. That's the biomimetic gel that is provided by the 2-photon manufacturer. We were the first user working with this material. So we started by looking at cytocompatibility. Do the cells grow on this? What happens? Can we, depending on how much we expose, etc., how does that affect the cell growth? So Federico just made a very simple microfluidic device where you could then print different structures inside and grow the cells inside and then do microscopy and everything through. And I think this is one of the really good features of this approach, because you print it onto a glass slide, cover slip, so you can really do your microscopy just straight into the systems. And you can see that cells grow very well, doesn't compare much with or doesn't differ from the glass controls. You can both grow then endothelial cells, 2D, and then fibroblasts, which is what we wanted to put outside in the 3D environment. It grows also very well. When you use 2-photon, you get sort of a cross-linking gradient because it's in the voxel that you cross-link. So if you really, really zoom in on these structures, you have this sort of mesh structure inside. And that's something that we can see, sort of cross-linking. And then we were thinking, how does that also then affect diffusion? Because if we want to use the material as a barrier, and then we want to culture cells, the material should not prevent any diffusion, of course, because then that's what the cells should be doing. So we see that the gel is permeable to molecules that are smaller than 250 kilodalton, which we think is fine because that's for the growth factors that we're interested in if you want to look at cell communication. But we were also a bit worried. So what then, if we now have formed a vessel in this gel and then we inject another gel outside, is that just going to diffuse through? Because those gel precursors are also that smaller than that. And they're just going to diffuse through and clog the whole system. But Jacob found a trick to avoid that. So, which is a matter of sort of just basically in the order that you add and remove material. In the end, it is possible to make this on-chip vessel model. And this is just a different sort of schematic of that image I showed you before, which was more cross-sectional. So we have a microfluidic device. We have inlet here with an area that connects to an outlet here. And then there's a second area that surrounds that. So it starts by filling everything with a hydrogel precursor, then prints the structure that we want to use as a scaffold so that that connects these areas. Then we can remove the non-exposed and introduce the 3D cell culture matrix instead surrounding it, which then includes fibroblasts. And the really cool thing with this, so this is now confocal stacks, images showing this. What you see in blue is the hydrogel structure. So first of all, we define this before we put any cells in there. So we don't have any side effects of some cells being scarred or necrotic or etc. So we only have alive cells. The thickness of this wall is just 40 micrometers. So it's very, very thin. He started by like 200 to make it stable, but we push it down to 40. So that means that we're now really talking about communication between these different cells inside and then outside. This system, because it connects to a larger inlet port on top and bottom, can be perfused and perfuse it after the 2 weeks, and that's sort of stopped because we said, okay, it's not leaking. And perfused in a very controlled way so that we can really know how much we're putting in. And because of the high resolution, we can also define some complex structures. The angiogenesis process is super nice. It's spontaneous in all forms, but you don't know really in which directions your vessels will go. You don't know how many vessels you will have. So here we actually gain control of where are our vessels. And you can see, I think you can see at least here, we have the fibroblasts in the chest. We observed that there seems to be after a week or so, there seems to be more fibroblasts close to the vessel structure. Whether that is because they are actually migrating towards the nutrient source or not, we don't know, or whether they're just proliferating more there. But we can co-culture the UVEX with the fibroblast, 40 micrometer vessel. We can perfuse it, and I think that's really a really big benefit of this system compared to many other vessel models that you can see. And as I said, the fibroblasts seem to increase. So with that, I want to thank you for your attention. Give acknowledgements to the team and actually Federico was busy preparing for his defense, so he couldn't join when we took this group picture. I will share you the link for his defense if you're interested. And he's had a paper that has just been accepted actually in Advanced Materials Technologies. So it's out. So thank you very much, and I'm happy to take more questions.
So thank you very much, Maria. I think we have also room for one or two questions here. So I open up the floor for questions.
I have maybe one to start with. Maybe I missed it. Which hydrogel are you using for the two-focus study?
So with this one, it's called BioPrinting. We don't actually know what it is, but we think it's gel.
Okay.
Yeah.
Because you don't see even those during those 10 days. Do you see remodeling happening? Do you see that the cells coming out of the channel trying to go to the fibroblasts or vice versa? Or
yeah, we have seen things like that. Yes, we see protrusions, but I didn't include anything because we don't really know what it is we're seeing yet.
Okay.
Yeah. But we do see interaction for sure. Also with the two photon, you can control how much you expose, which in turn is how much you cross-link. And that is also something that we see also connects with the cell interactions. And something that we're studying in more detail. It looks
really stable.
Yeah.
So that's good. If you want to pick it for a long period of time, right? But then there's always the caps. If it's a gelatin-based, then gradually the cells will probably start to degrade it, especially in the fibroblasts. So then, the long term might be a problem. It's interesting to see that with the photon they have that high cross-link.
Okay, thank you very much, and thanks everyone who has asked questions now during the session so far. We will now close the workshop for a short lunch break. We're running a little bit behind schedule, so we'll push the start to 1:01. 10 past. Sorry. So in roughly 35 minutes we will return.
Perfect.
Thank you, everyone.
So hopefully you can see now my presentation. Yes. Excellent. So it has been, well, thank you first of all for inviting me. This has been a very interesting day to listen to you all. Unfortunately, I won't mention the kidney in any slide of my presentation. So this presentation will be from the other perspective, from the regulatory agency's perspective on new methods. But hopefully this will be some information that will be of use for you, I think, in the future. Maybe not right away, but hopefully in the future, because I think we have the same goal, to improve the methods to better predict efficacy of safety of new medicines. But to give you a little, for those who don't know what we do at the medicines agencies around in Europe. Well, the first statement is taken right from our webpage, and it sounds a little bit fancy, but it's to ensure the quality, safety, and efficacy of medicinal products to safeguard public health, public and as well as animal health. But it's also about supporting innovation. So that's why I'm very happy to be here today. Well, we don't act locally at the national level, but mainly at the European and international level. So therefore, it's also important to get everyone around the world on the same page with regards to new methods. But we work by doing the assessment of dossiers
before entering into clinical trials, as well as applying for marketing authorizations. And we also support development by providing scientific advice, both nationally and at the European level. But I guess you're familiar with that the pharmaceutical area is surrounded by very strict regulations and requirements regarding what is needed to support efficacy and safety. And we have EU directives and soon also a European regulation that is implemented in the Swedish law. And we are also, of course, following the Directive 2010/63/EU, which is all about the 3 Rs. And although what we are mainly familiar with are in vivo methods, we are of course aware that there are a large number of animals used in research, of which a significant part comes from use for regulatory testing. But as I listen today, I think what you mainly are working with now is providing new methods for both basic research and translational research, but hopefully these will also be sort of available for regulatory testing in the future, so we can all help reducing the number of animals used in regulatory testing. And we also are very happy to see that progress is being made, in particular in early drug development, but perhaps before entering clinical trial and drug development in clinical trials. So where are we today? Well, as I said, we note that there are already alternative methods to in vivo testing available, in particularly in early drug development phases. Both cell-based and in silico-based prediction methods, and also for exploratory safety testing, and also whenever there are safety findings in vivo studies, in vitro testing or alternative testing can be used for understanding better the mechanism behind this toxicity and the clinical relevance. But when it comes to regulatory testing, with that I mean mainly safety testing and non-clinical or quality control. Here we work mainly with trying to reduce and refine the use of animals. Because for regulatory safety testing, it will require very complex model. And we also need qualification of this test for replacing particularly some very complex in vivo models. So just to give you sort of a sense of what the regulatory testing requirements are for you who aren't familiar with the requirements for drugs in particular before entering into the first clinical trial. Well, it is still heavily relying on in vivo testing in animals, and it's also very these are legal requirements, but of course exceptions can be made if there are scientific justifications for that. But testing is made in a very broad sense because we don't really know. We want to ensure, of course, that the substance has an acceptable safety profile before going into humans, and we need a very broad testing. We can't really know which type of safety issues will emerge. Of course, for some specific type of drugs, such as some biologicals like monoclonal antibodies, which are very specific, which acts on very specific targets, we can usually have a nice sort of understanding of what the safety issues will be, because they are quite related to the pharmacology of the target. But for small molecules, we do still need this quite strict testing. And non-clinical safety testing is done to answer some important questions that we need to have before entering into clinical trial. We need support for the efficacy and to set a dose level in the clinical trial. And I think from what I hear today, I can see, of course, that the models that you are developing could hopefully be used in this part of the drug development phase, where we now see different types of both in vitro and in vivo testing. And here it's very important to note that although it's expected that this type of data will be available to support entering into a clinical trial, there are no specific non-clinical guidance on exactly what type of requirements that need to be met. And this, of course, is depending on that there are so many different diseases and disease models. So here it's up to the development to justify the type of methods that are used to support efficacy and to show that these are have an acceptable standardization and are able to provide to answer the specific question at hand. And then, of course, we need information about uptake and distribution and fate of the substance, and to understand if there are any metabolites that could be a safety concern. And the typical, and what I think of as a regulator as the most critical data is the safety data that comes from repeat dose toxicity studies that are performed currently in 2 different species. Because this is supposed to answer what exposure is safe and what are the potential risks, both of the short-term and long-term use, and to understand if the effects we see, if they are reversible and possible to monitor in the clinical trial. And of course, non-clinical data is of most importance before entering into humans. But even with clinical experience, there are some data that we can obtain usually from clinical trial because of ethical reasons, for example, such as data on carcinogenic potential and risk for effects on the fetus if we are to expose a pregnant woman. So for all these, to answer these questions, we still rely on the in vivo models. But of course, we are doing what we can when we are providing scientific advice and when we assess both clinical trials and marketing authorizations application. We're doing what we can to reduce the use of animals and to sort of support the development of new methods. And the main sort of scene where we do this is when we work together with other regions in the world to develop and update our guidelines. And I just would like to mention that we do see some areas where we actually see some replacement of in vivo studies by using stepwise approaches, for example, where we start by using vitro methods. One example is the test of phototoxicity. And in some recent updates, the 3 R's and development of new approach methodologies are mentioned. And one is the reproductive toxicity testing guideline, the ICH S5, where we have a special annex where examples are given on and reference substances are being made for developers of new approach testing methodologies. However, so far we haven't seen any development in this area, although this is specifically encouraged by this new revision of this guideline. And we also have quality batch controlled guidelines, or for this we have the European Pharmacopoeia monographs, which specifies which type of testing is needed to test a batch of a drug that is on the market. And here we have seen some recent developments of alternative methods that are promoted, and we hope to see more use of these methods in the future. But as I said, we are mainly working with reducing and trying to reduce and refine. And one such example is the guideline that we have for biologics, where there is no specific requirements for testing in 2 species. And it's strongly enforced that you should only test if the species is pharmacologically relevant. So 2 species are not always required. And the same goes for a new revision of the carcinogenicity testing guideline, where alternatives are being made or proposed, where testing in 1 species could be acceptable. And we also try to refine the current guidelines or the use of animals by promoting that you could address several different aspects in one study. So you introduce endpoints, so you don't need specific testing for a safety pharmacology, for example. It can be incorporated in specific cases in repeat dose toxicity studies. And we are also doing what we can to promote trials in scientific advices, as I said. If we don't see that a test will provide relevant information, we discourage the performance of such studies. What we do not currently do at the national level is provide advice regarding new approach methodologies. And this is because this is highly sort of needs specific expertise. And as you may understand, because we are still working with in vivo models, these are the main areas where we have expertise in. But to provide support for development of alternative methods, this is covered by at the European level by the European Medicines Agency. So in the last part of this talk, I would like to sort of give you some information on some very useful information on how the European Medicines Agency could support development of NALs. EMA has in their EMA regulatory science documents it's a strategy document that stretches to 2025. It has specifically pointed out enforcement of 3Rs in this document to raise awareness on 3Rs and new approach methodologies. And to provide support in different ways, both to provide some general recommendations to leverage and qualify 3R methods. And they have developed, to jump to the last point, a specific group for 3Rs. It has been active for some years, but there's been sort of a regroup and with new members in this working party to work with these specific questions. And this is a highly active working group that engage with stakeholders. And the idea is to both engage with all stakeholders and also with the European regulators network, because a lot of training is needed, of course, to meet sort of the need out there, because we do see, and I'm very glad to see that there are so many new methods coming, and there's a sort of strong wave of development in this area. And what EMA can do is also to provide qualification advice if you have advanced far enough to have a validated method and you want to reach regulatory acceptance. And recently, the Innovation Task Force, which is a specific group within the European Medicines Agency to support innovation, they now offer a very early dialogue on 3Rs, on how to proceed to reach regulatory acceptance. And EMA also have a guideline, which I recommend you to read. It also provides general principles of regulatory acceptance of 3Rs, and it provides some criteria. It's a step-by-step approach, which is very good for you to read. But what I can say is that it all depends on the context of use of your methods. Of course, even though if you're developing a method that would be used for better understanding a disease, or you will use it for an efficacy model, or whether you are going all the way to have this regulatory accepted and try to introduce it into international guideline, you can always reach out to the European Medicines Agency to seek qualification of your method. But this provides sort of a very broad advice on what you need to think about. And just to, well, here, of course, I have given some routes if you want to know more about the specific routes for either approaching the European Medicines Agency if you seek an early dialogue, or if you have come more far and you want to qualify your method. This slide provides you with some information, but it's also very easy accessible if you go to the EMA website and search for, you can search for 3Rs, for example, or qualification, and I think you will find this information. And for the sake of time, I think I will actually stop there, because I think this last part of the meeting with the discussion and interaction, I think we should give some room for that. But with that, I say thank you very much again for inviting me, and hopefully there will be an interesting discussion now that follows.
So thank you very much, Camilla. I think we will we have time for some specific questions for Camilla before we move into the panel discussion. So I leave the floor open for this for questions from the audience, from people here on site, and also for people who are participating through Zoom. Maria?
Yeah, I would like to ask you a question, Camilla. Thank you very much for your presentation. How much is the Swedish Läkemedelsverket affected by changes on regulatory aspects in other countries? I'm thinking, for example, what the FDA is doing now, was previously mentioned, of opening up for organs on chip models.
Well, it's very both easy and complicated to answer that question, because I think already now we are quite open to alternatives, because we do see some applications where it's not possible at all to do in vivo studies because they're not relevant at all. So it is the sort of relying on in vitro data. So we're not unfamiliar to that, and perhaps we are not as strict as the FDA in following the international guidelines. We are more pragmatic in that sense. And what the FDA is doing so I could also add that I'm a participant in this joint 3R working party at the EMA, and we do have interactions with FDA where we sort of exchange experiences with, for example, organ on chip models. But as far as it goes for organ on chip, I mean, companies or developers are welcome to use that, and it's not problematic as I see it if you use it to support efficacy or mechanistic studies or in early exploratory trials. It's when it comes to the regulatory testing where we need agreement on what the standards should be and are encouraging qualification advice at the EMA level. But we don't see that it would be a problem to use it if it's validated and a good justification. So it's not. I don't see it would be problematic to use it to support.
So maybe I can just rephrase how far is Sweden from using organs on chip for regulatory aspects?
But we don't act by ourselves. So I can't answer for Sweden. We work at the European level. So we can't have any different views than at the European, because applications are now done at sort of a joint level, also the clinical trials with the new clinical trial regulation. So I can't give sort of a Swedish perspective, but we.
No, but maybe you're. Or yeah, following FDA.
Yeah, just a half follow-up, just because it's very interesting to hear what elements the focus is on, specifically which is of greatest focus. But you were also mentioning that these, let's say, more advanced microphysiological systems can be used to determine efficacy and everything else, but this seems to be heavily focused on people using them and demonstrating them. But are there any efforts of the medical agencies to try to quantify some of these data to say, yes, we can now approve this as a system for doing X, Y, or Z treatment actually during the clinical stage, because we've seen others doing it? Because from from what it sounds like, it seems to be heavily weighted on people using it themselves and validating themselves to show that this is working. But the medical agencies are still going to leverage on their more established technology because there's no motivation for them to actually do it. So I suppose the question is more, are there committees or panels or internal EMA entities which are actually trying to benchmark some of the more advanced microphysiological systems against established, in vivo models?
Well, not to my knowledge. There are no such projects as far as I know. Well, but of course, we're interacting with the developers. Yeah.
But wouldn't that be seen as like one of the critical bottlenecks then of all the 3R things, is that it's seen as this is perfectly great, and it's like, as a community in general, we encourage people to do it, but then it has to be taken by the cost and everything else, but it can never actually be used in the regulatory space because none of the regulators are actually benchmarking to see if it can be used.
Exactly. What was my question?
But as a general response, it's not for the regulatory agency to drive development. We can support development in the ways that I described. But I see your point that more interaction is needed, and I think at an earlier stage than now, so that methods are developed that could fulfill the requirements of the regulators. And I think that's why it's important that the general 3R work that is done by the EMA. I think I'm hoping that we will be able to provide that kind of guidance, more specific guidance, and collaboration will for sure be required, in particular to be able to, in the future, to replace the larger models, the standard toxicity testing, for example, because that will require assessment of a large number of endpoints or answer a lot. And for that, we need sort of a combination of techniques and also AI, of course, to analyze them.
Yes. So to start off, I want to send a question to Carlos, perhaps, just from the discussions today that we've had. How would you summarize the insights from today, and what does this mean? What we've discussed today for the BIRDIE project? We've heard a lot about different kinds of models, and also some of how in vitro models can be used and cannot be used in a broader context today. So what are the sort of important insights for BIRDIE?
Well, first and foremost, I want to thank all the speakers for making time to participate in the workshop. It's quite useful for us to have different stakeholders at the table. So we had AstraZeneca, more looking at the perspective of end users, but also developers and researchers on using microphysiological models. We have colleagues also working on development of different aspects of microphysiological models. So it's very insightful for us. And we also have then Camilla, in the end, part of how we can make use of the microphysiological models, then hopefully helping out on reducing 3R, or eventually in the future, putting a more promising perspective, replacing animal studies. It's clear that we are far from being able to do that. There's a couple of indications, more in the US, that it's possible in some specific applications that it's possible to demonstrate that the microphysiological models are able to benchmark the safety of compounds. So things are moving, and they are moving really fast. But it's important, and it's clear that we have to improve on communication between all the stakeholders. And this was also noticeable here today. It's also more important to be able to speak the same language and to have. I mean, the interest is there, the clear target is there, but then there's a lot of bits and pieces that need to be well connected. So with that, I would like to thank you all to share your perspectives. It's very insightful, I think, for all the partners of the project that are sitting here, mainly the PIs, but also the researchers online that are doing the work in the lab. It's very insightful information. And it's also good to see that we all have struggles and different aspects that gradually we are trying to mitigate. So that being said, it's clear that we need more advanced models or models that are able to answer our research questions, of course. So that being said, I think we did have a good startup with the presentation. So we'll leave the floor to you for the next questions.
Yeah, so I want to follow up with a sort of very broad questions where you can provide your kind of specific perspective on this. Why is this interesting at all? And I mean, what do you see as the key benefits of these new advanced in vitro models, not only in terms of replacing animal models, but also over conventional 2D cell models? What are the key benefits here? So perhaps I can send the question to the whole panel. Silvia, if you want to start to provide your perspective on this.
Yes, of course. I think you already mentioned there is a big drive towards alternatives to animal experimentation. But the question is, should this be the only drive, right? Are we making things too complicated for the sake of checking a box? Or there are some situations, research questions, where 2D is still okay, right? So are we overcomplicating where there is not a need for that, just to be in the trend? And that will be one of the discussions that we usually have in our laboratory. But for instance, we are in those studies where we are looking at transepithelial transport, for instance, in our case, definitely we cannot do this in 2D. So there is absolute need to create conditions in which we can assess this function. And that cannot be done in 2D. So for instance, this is an example that drives us to look at microphysiological system as well.
So do we have a follow-up comment on what Silvia has been saying? So Maria, if you have...
I mean, so the thing that motivates me to do research in the area is really the belief that we can find, new things that we cannot find in vivo models. And I think this is interesting from a research perspective as well, that if you can isolate events in a more controlled fashion, we can learn about disease mechanisms and so. So I come in with a more like basic science motivation to be able to dissect the animal and not have all of these interfering effects from all other organs, etc. To be able to simplify and look at specific questions. That's one of the things that drives me in doing this tech development. And if that also then can be used in industry, to cut costs and make sure that more medications reach the society, that's of course great. And if we can clear out the ethical aspects of animal use, that's fantastic. But I think it provides sort of platform to make interesting basic science studies in a controlled manner.
So then I'm from your perspectives, Emelie and Philip. Where do you how do you see kind of new in vitro models as interesting?
I think the translatability aspect is is also very interesting and that's the aim that we have too, is to look at disease models with maybe cells from patients who this is relevant for, and to see them in not just in a 2D culture, but a more advanced system where we can actually, for the proximal tubule, look at what's happening basolaterally and apically, and to be more physiologically relevant. I think that's what's offered in part by these models.
Essentially to have models for what questions we're facing. Because just as Silvia touched upon, in some cases it can be good enough to use conventional 2D systems, whereas in others you lose out on a lot of the actual physiologically important mechanisms and functions that you need to study, both from the perspective of utility in drug development as well as from the more research and basic science perspective. There is a lot that is lost in some cases if you stick to the conventional models. So essentially having access to both gives you the toolkit you need to answer different types of questions, all of which need to be answered to drive the research forward.
So I would say perhaps as a researcher, it's quite easy to sort of fall in love with your methods in a way, and to see kind of an inherent value in what you're developing. But Camilla, if I look at your perspective, I guess from your point of view, what the actual model is not as interesting as what it can do and how it can sort of recreate biology in a way. Is that a fair representation of your perspective on these new kinds of in vitro models?
Yeah, I think you have made a fair conclusion. But I have to say there are instances where, for example, a drug is put on the market and there are some safety issues emerging, and you are thinking about, is this related to the drug or not? And you haven't seen it at all in non-clinical species. I mean, if you have a validated model that could address this specific safety issues, you could address it in this type of model instead of trying to investigate it in human, like the probability that it would be treatment related. So I mean, I can also see that the value of these type of human cell models or organ-on-chip models for sure.
Thank you, everyone, for your comments here. I'm going to move on to the. Oh, this should be a 3. So, what would you say is sort of key success factors for transitioning these kind of new type of methods from, I mean, when taking them from the fundamental research to actual is very perhaps provocative, because the fundamental research is also actual, but to a broader applications, not within just the isolated research context. So I mean, perhaps Emelie and Philip, that's perhaps a question first and foremost directed to you, because that's what you're interested in.
Yeah.
Anyways.
I would say, as I also touched upon in the presentation, that it is to what extent they are adapted for the different types of assays and endpoints that we need to study in order to actually assess mode of action or safety, efficacy, depending on also in which stage in our work that the chips are implemented. So essentially, it needs to not only produce a model system and an environment that is physiologically relevant enough for the particular aspect that you're studying, but also to do so in a way where we can actually assess it in the ways we need.
And I think looking forward, whether we can use these as, if they have a predictive value, so if we can manipulate them or perturbate them and actually get sort of a prediction of what's going to happen in the clinic when this is going on. So there, that sort of intermediate there, I think that's where we need maybe mathematical modeling, AI as well. But I think that's a big deal.
For sure. And sort of the phrasing of this question, perhaps Maria, is kind of, it was a bit pointed toward an intentional kind of provocation there. But how do you see this question in terms of taking what you are developing within your lab and kind of seeking a broader application?
So, I mean, I think this is very important to address, because if we do a lot of, tech development or basic research and no one can use it, then what was the point, basically? It could be fun, it could be interesting, and maybe in the future it could be used for something. So, but I think actually what we're doing now, so we've just finished one big project that was running for 5 years, and that was okay. So now we understand how this tech development works. We know what we can do, what we cannot do. So now we're ready to take the next step and say, okay, if someone's going to use that, what are their needs? Because I think it's also very difficult to find exactly the right spot to interact with an end user. Because if we were going to Astra 5 years ago and say, oh, we want to make a device, they want to use it, of course, and say, oh, yeah, we just need 5 years first to make it. And, of course, sometimes you can have these long-term investments and work together along that path. But you also need to be at a stage where you can say, okay, we could actually start making trials right now. So I think as a researcher, the biggest challenge is really to find the right time point to address end users so that there is a common interest on both sides, where it's also interesting for more research and it's also interesting to use. And usually you should start the discussions as early as possible so that you develop together. But if it also takes a very long time to develop, it's risky that you lose interest. So I think this is a challenge of finding the time when it's fun or interesting on both sides.
So, I mean, I want to dig a little bit deeper in that perspective. How do you find these collaborations between the kind of developer and the end user, and how do you cultivate them? Carlos, if you want to connect.
So the best thing is to work together, but then there has to be a clear interest, right? And I think here the main bottleneck is that on the side of the end user, I see a huge wish list and something that works for all the fundamental questions that they might have or research questions that they might have. And the other side, there's still a bit of more fundamental research that is needed to fulfill those wish list points. So it is important to have all the stakeholders at play, even if it's a lengthy project. And it's also important to involve stakeholders. As we mentioned, Camilla, it would be important to have EMA or FDA more engaged. I know that there are societies that are doing a bit of that are trying to bundle more researchers, more companies to then interface with regulators, to also try to understand how to bring new technology to the regulatory space. But that will take time. But I think it will be. I think we have to build a niche tree that we have built for the 2D models that took decades. Of course, that we would wish that for the 3D models and microphysiological systems would be faster. Hopefully, it will be faster, because otherwise, we are going to be stuck saying that we want to replace animal models. On the other side, we are developing even more fancier in vitro models, but then we have a roadblock. We have a death valley between the two things. So I think, having all the stakeholders clearly thinking throughout the path and clearly engaged, it's essential. And this is an example where we see, that this is more and more essential, of course. But both will be important. Of course, that to manage a very big wish list with very big fundamental research will take years for sure.
Yes. So if I bring you in this conversation, Camilla, as well, where do you see? What's your kind of ideal phase or stage where you would kind of get involved with emerging technologies and collaborations between kind of researchers and developers and potential end users?
Well, as I see it, as soon as you have a clear idea that this could be something that could be used in regulatory testing or would be important in drug development, you should approach, either seek a partner in a pharmaceutical company that could use this test in parallel with the standard testing, and also seek an early dialogue at the EMA. I really think it's good to have this early dialogue, and you can get advice on additional reference substances or additional steps that you need to take. So I really encourage an early development. And with that said, I would like to thank you all. I will have to actually leave for a meeting at the EMA, so it has been very nice to participate in this meeting, and I would take it with me. I hope to see you again soon.
So,
yeah.
Thank you very much, Camilla. Is there one last kind of message that you would like to send to the audience?
No, I can see that much is going on, and it has really made me happy to listen in today. It's very advanced. So I hope to see it applied. Thank you very much.
Thank you very much for participating. Silvia, I also want to bring you into this conversation. So, I mean, Maria mentioned sort of having fun in collaboration. Is this something that resonates with you as well? Or how do you form kind of collaborations with other researchers and end users and other stakeholders?
Yeah, I think one of the aspects we need to take in mind is that usually PhD students are excited about projects and they want the projects to be as sexy as possible. And then the project is done, and then we move to a next one to discover the next new thing. And many times for this transition, you need to sit a little bit on what you have found and try to cover all angles of it. So for that, we have, for instance, these big projects on assessment of chemical safety, pollution, which might not be the most sexy projects, because it's a lot of screening and a lot of assay development and optimization and validation. For instance, for key events, what are the key events we are going to look at? And then imagine that now Carlos is looking at other assays, uses other assays. We will never be able to compare our information, never. So sometimes from the researcher perspective, you want to be as unique as possible, do unique things as possible. Not what Carlos is doing, for instance. I'm talking about Carlos because we are in the same country, and it's pretty small. But I think one of the key success factor is also that we need to align. And if we test, for instance, the reference compounds, that they are the same reference compounds at the same concentration, so that we can move on rather than just reinvent the wheel every time with. So I think this is also an incentive that we give for our researchers, because we don't want PhD theses to sit in a library in a nice booklet and remain there. We really want to have an impact, because that's what we also write in the theses. We want to make an impact in human health, and we cannot do it just by stopping at publication. And as I mentioned, we are putting ourselves in contact with people where we can learn. For instance, with AstraZeneca, we want to know what is needed. So we are open to the dialogue, and we are open to this refinement. Please tell us what are the reference compounds we should test, so that we can do it. And I think it's we are very open and receptive to ideas and suggestions, of course.
Yeah, so I have I think very relevant question from the audience here as well. about what's the role of research funding agencies in all of this in terms of advancing new like in vitro models, non-animal models, and they also and also the comment there they mentioned this first call for non-animal models in Horizon Europe now. So what's your perspective on funding agencies and their role in sort of bringing this forward into implementation? Maria, do you want to?
And I was actually thinking along the lines before we spoke, and I think it was, I can't remember what it was you said, Carlos, but it was something about, I think motivation is the word you used. Yeah. I mean, because there must also, of course, be resources. You know, we can all want to work together. Timing can be perfect, but someone should also do it, and that person probably wants a salary. So I think that because it's so, just speaking now from Sweden, the Vinnova, which is the innovation agency that we have for funding, where you would normally go for this type of project. They typically give quite short projects and with limited funding. And I think that is an issue that we have. That, so there were just some new grants that were received, and it's a 1 year. No? And what can you do in 1 year? Then you really must have a device that is fully equipped. It's just to interface. And that's, we want to start working together earlier. So I think that's a challenge. And I see it very positive with the new EU call that you just mentioned, with alternatives. Hopefully that can be a more long-term one. Because if you would then either go to BR, for example, for funding rate, which is for a couple of years, then it's more on basic science and fundamental studies, and then it cannot be too applied. And so I think that is something in general in biomedical engineering in Sweden that has already been identified as a bottleneck. How can we take new technologies into life science industry or hospitals, or who shall support that transition? I think that's a challenge that we have nationally.
Any further comments on that?
Well, I think the European Commission is making also an effort to connect the dots, right? I mean, historically, there have been funded projects on multiple things and repetitions of funded projects on similar topics. So the Commission has clearly tried to implement ways to bring these projects together, especially the projects that have been funded in similar funding rounds and in similar topics. So there is an effort, but it's very unlikely and very difficult to have all the key stakeholders on those projects due to the nature of the funding. So what we see more and more in the specific call, I think they mentioned that, if it would be in the realm of the GRC, that the GRC would be involved. So there will be different stakeholders that the Commission is also trying to involve as, research laboratories managed by the Commission. But I still feel that there is, the regulators' perspective that is still not there, the patients that is still not there. I mean, we are asked to bring patients, and of course that we would like to open a dialogue with patients, but it's not that it will be possible for us to reach everyone. So then we normally involve patient associations, and then they will interface with the patients, of course, but then we will not have the same degree of information. So that the problem is a bit more substantial than what we can discuss here. But, these small initiatives are important because it will address a lot of fundamental research, but then having more the perspective of the actual application would be also important that the Commission makes it in a bit of a more integrated way. So we see dynamics and we see change happening, but we see also a lot of national initiatives. So also in the Netherlands, we have quite a lot of different national initiatives now on regenerative medicine, on in vitro models and so on. So I also see now, different countries trying to push it with the internal funding, which is nice, but, we are still Europe, so we're not putting all those initiatives that are a bit satellite now together to push this through, because there has been a lot of money invested on these initiatives.
move on. I think, yes, I think with this, is there something else that you would like to add, Philip, towards this question, how we should kind of implement new in vitro models in pharmaceutical research?
I just want to say that we recognize the different sort of incentives and what Silvia mentioned, that incentive to be novel and to develop something new, which is of course what we need, but also from the pharma side, we do need something that we know will work and be reproducible and
consolidated. We don't want to be really
so there's a bit, I think more dialogue certainly will be needed there. But yeah, we recognize that there is a difference in sort of incentives from these two sides. I don't know, what would you say more to this question?
Not much more to add really beyond what's been discussed already.
Yeah, I think we're good. Yes, so I can bring Gavin here.
It's going on the side. No, but just to tie those two things together, I mean, from sitting on both sides of the fence for quite a few years, it seems that that's one of the missing issues. Like, you can't get a research grant to do the boring comparison of something for 3 years on the same thing to get validation data so that someone can actually use it. I mean, so on one aspect, there's money and opportunities to get funding so that you can try something new. But then once it's been tried to do something new, the 4 years of testing to show and actually have somebody use it, there's no money to get someone to do that part. So then there's this gap in the middle of, well, who funds that bit? I mean, a lot of times that is left up to the development companies and other things, but as we were touching on with Vinnova and other things, a lot of times these are only partially funded programs. These is like 50% at most funded. And these are generally really expensive programs where, I mean, it does take, 3, 4, 5 years to actually start getting, repeatable data and things. And I think that's one of the kind of biggest roadblocks that we see right now, is that you can have amazingly new technology, but actually getting market-ready validation of enough screened compounds and enough, let's say, collaborative efforts to demonstrate the true utility of a platform isn't really there right now.
Yeah, and I mean, especially if you wanted to sort of have your model validated in different labs and tested and compared to sort of really benchmark something, then that requires a great deal of work. That's sort of left, as you say, in between in many ways.
Yeah, but I think also the incentive in pharma industry that's come a little bit more recently, like to publish more. I think to have more collaborations with academia, like we had in the 3D-VAS PT. There is a bit more incentives to do that, do that kind of work, and maybe also characterize and see what is the maybe do an NGS or like see how does this method compare to the previous methods, and then share those results. I think that's at least helpful along these lines. That's what I do.
Yes, so I mean, now the discussion has sort of, we've broadened the discussion a bit, and now I want to sort of bring it a little bit back to where this whole thing started in a way, about what are the particular challenges associated with sort of modeling kidney function and kidney disease. And I think, I mean, if you want to start here, Silvia, I mean, you've touched upon a lot of these topics already, but to give kind of a condensed version of what the main challenges are.
She's anticipating a lot of challenges.
I think it's always very important to know exactly what is the context in which or that I want to mimic. What exactly do I want to mimic? We cannot have it all. The kidney is a very complex organ. There are more than 20 cell types. We cannot have it all. So many times when we have the discussions in the lab, when we say we do a proximal tubule on a chip, we are now starting to say we do transepithelial transport on a chip or reabsorption on a chip. So focusing more on the function rather than the architectural element of the kidney, because we cannot mimic the function of the proximal tubule in totality. right? So that is what we are now, one of the major challenges is to really refine what our research question is and not try to do everything at once, because it will not be possible. So one function at a time, and depending on that function that we are having in focus, all the design of the chip or of the system changes, the experimental setup changes, and so on. But then it comes back to these discussions of, shouldn't we have a model that is easily adaptable, that we can put other function in the same chip so that we don't have to redo another model. So it's all these discussions are very difficult. So we try to be as simple as possible, but still retain some complexity in the models, which is very difficult to do.
It's a very fine balance.
So I mean, it sounds like part of the sort of the goal is to make things modular in a way so that you can connect the specifics with the general. And any further comment? What's I mean, from your perspective at Seneca, what do you see as particularly challenging?
I'm not well enough versed in how much disease modeling we've actually done with our chips. And like in the work that I do myself, is more studying a function in healthy cells rather than how that is impacted and affected in disease and how best to mimic that. But I would imagine, like, the better you can mimic a function in your culture, the better you can mimic taking it away, and then study disease models, either onset or cure that way. But I don't have much more than that, I'm afraid.
I think what we're seeing, what you've touched upon already, that the interconnectivity of the kidney, like for instance, we know that inhibiting SGLT2s will also improve glomerular function, but why is that? And so the more complex questions are a little bit difficult to model, and we're talking about that also in different projects. Like, why do we see this proteinuria in this model? We can't really explain it just by modeling the filtration barrier, just by modeling the tubule, and also like recapturing like things like tubular glomerular feedback is also a big challenge. So just I see the advantage of focusing on a question in one system, but often, and especially the drivers of kidney disease are often not only related to the kidney, so it can be diabetes or hypertension, and that's also making it more difficult to model with specific cell types.
But in a way, knowing these things from the in vivo case can, in a way, serve as a validation metric as we begin to expand the complexity of the in vivo systems, right? Like, to what extent can we then model and maybe can recapture these effects that we still don't fully know why they behave this way, but that they behave this way? To what extent we can start to mimic that as the systems begin to grow more complex, right? At least that's kind of something to reach for in the future.
Well, it all starts with a very simple research question, but then one research question leads to 10 other research questions, and you have to build on the complexity. And I think here the main, we are looking just at the model, the in vitro, but the cells will play a key role. And if now we are using immortalized cells, we are using iPSCs from healthy donors, but what is a healthy donor? We never know, right? So, and we have generated iPSCs from a donor with a specific disease, but what is the disease stage? So, the mission is, of course, that we can oversee all of this, but it's it's still not there yet. But that's in the context of kidney and in the context of the nephron, which again the 20 different cell types, they are working together. They have tons of different function, tons of different crosstalk, and they will behave totally different when they are exposed to a compound. That complexity would ideally need to be there, but it's if we put all there, then the readout it will be extremely complex. The techniques that we have nowadays developed will not allow to have a complex readout. So all of these things need to mature at a certain extent much more.
Yeah, and I mean the animal models are not really there yet either.
Yeah.
Not so quite seriously lacking, but of course we need something also that's good enough. It doesn't need to be perfect.
Yeah, I think that's I think a good perspective. I mean, we don't have to chase what is perfect. It just have to be good enough and better than the next best alternative. So we're starting to sort of get close to the end here. I think, I mean, this is perhaps a message or a question to those who have been through the kind of the development cycle, and also, I mean, perhaps would be good to have Camilla's perspective on perhaps, but what are the pitfalls that you need to be aware of when developing new models? Do you have previous success stories and failures to share? I send this question to the whole panel, perhaps.
Yes, Silvia.
Yes, Silvia.
I think I have a successful failure story. Rather.
That's the best way to start.
That's the best way to start. Now, I think it's very important when we develop, we come models, we come with ideas, and then we put this, and then we perfuse from there. And then it turns out that we have to manipulate a lot by our hands. That so the reproducibility of what we have just designed between different users, it's not there. And we try to tweak in different ways, and then we come up with discussion of the secret leprechaun in the lab that might destroy the experiments during the night because we just don't have an explanation. So that's why we now try to make things as easy as possible. And as it was mentioned, it doesn't have to be perfect as long as it works, and it's easy to reproduce from day to day, from user to user. And then if we are working with cells, it can happen that the cells are not the same between passages. So that is also very important to make sure. They are very sensitive. Sometimes even taking them out of the incubator might affect them. So we try, that's why it gets to this point of standardization of our procedures, which researchers in an academic setup are not very keen on having standard SOPs and so on. So we try to bring this mindset of we need to standardize our procedures so that it makes our life easier eventually on the long run. So that was what we learned from our successful failures.
Yeah, it's also noticeably a limiting factor that no one shares the pitfalls. I mean, we don't publish pitfalls, we don't publish negative results, and those are also extremely important, especially when it comes to implementation and to make sure that the technology is robust. You know, if you might have a model that is working in your hands, and then you transfer it to another lab, and it doesn't work the first time, the second time, and the third time, because you never shared eventually the pitfalls that you faced in the beginning. So that's also an important thing to keep in mind, and important to address as well. But it's difficult. No one likes to speak about the negative results.
And what I have noticed, and it's being published at the moment, are these studies of cross-lab studies. Okay, I developed this model and I send it to somewhere else, and this is what the result, and then the comparison between the labs, which I think is also very important as well. And it needs to have a stage for this kind of studies to be published. It will not go to, everybody wants high impact factor, but is that it matters, or is the impact on the development that is the most important, right? So I think there is a platform at the moment for this kind of studies, and we should explore it.
Yeah, very good perspective, I think. So we are getting close to the end here. I want to end on a question that sort of brings in another component here, because now we've talked a lot about interaction between researchers, developers, end users, and also regulatory bodies. But how can we as actors in this space, how can we also interact with a broader general public? And what are the kind of challenges in terms of bringing these topics to a broader discussion? This, I think, is a question for the whole panel. Do you have good examples, perhaps, of working with a broader public? Yes. So Silvia again.
Yes. Well, now I'm going to tell you about a successful story. A real successful one. So at the end of this month, there will be the Dutch Design Week, which brings together general audience. Yeah, I don't want to call them normal people, but the society, to see what exactly is happening in the area of design of, it can be anything. So we are going to be there with a kidney on chip project, and to show how basically kidneys are working. So we had to design a small prototype with fluorescent dye. So very bringing back to the basics, exactly what we want to mimic. So that it's easy to understand with UV light. So we bought some lamps so that people can shine light, and then there is a yellow dye with a red dye. In the idea of bringing awareness of why is it important to understand how organs are working, in this case the kidney, and how we can replicate their function in the lab without using animals. So I think for us in these kind of settings, like going to places where we will interact with the largest, the general public. It's also out of our comfort zone, because it also, we need to change our language at the time, but then it's also very interesting perspective, because then it gives you a different, it forces you to think a little bit about what exactly are we doing and why are we doing what we are doing. And I think this is what we are doing at the moment. We will go to the Dutch Design Week and we will show how kidneys are working and how we do it in the lab, how we model the functions in the lab without using animals. That's an example, for instance.
I think also what you mentioned, Carlos, bringing in patients, as you did in the former workshop, to actually show them what's being done in more fundamental research.
Yeah. So there's a caveat there, right? Because the patients are, you don't want to generate a false hope or immediate solution for the patient. So then you have to be, that's why foundations are kind of a middle or an intermediate. Making general audience aware and the general public aware is important. So I find it remarkable. If you ask anyone that you cross in the street, why we shouldn't use animals, they will probably answer you. But then if you ask, okay, what we should replace the animals with, they will not know how to answer you. So I think even, regulators and so on, they should also advertise that they are making an effort, or not even the regulators, even the European Commission, that they are making an effort on bringing alternatives. And these alternatives go and then try to explain in simpler terms what are the alternatives, because then people become a bit more familiar with the topic and what are the option, the plan B. So that's also important. It is important that we do it top bottom up, but it's also important that there is an effort also top down to make people more aware that that we are looking into alternatives and working into alternatives. But that it will still take some time that these alternatives will be able to replace totally animals. So if you speak with general audience, then everybody will say, okay, animals are no longer used in Europe to do preclinical or preclinical studies, but it's not true. It's still needed. It's still being requested, and just because of the fact that the alternatives are still being worked out and being optimized. to replace the animal models.
So I think that will have to be the sort of it's an opportunity and a challenge at the same time, and I think that's a good sort of point to close the discussion. I want to thank all the speakers for participating in this workshop. I learned a lot, and it was very interesting to see all the different perspectives on modeling kidneys and sort of approaching how to kind of mimic biology using technology in so many different and diverse ways. And I also want to thank the audience who has stayed with us the whole day, despite some technical hiccups along the road. So this presentation or this workshop has been recorded, so it will be made available through through BIRDIE's website, and we will also share it through our different social media. So keep an eye out for the recorded version. And thank you, and have a very good rest of your day.
Thank you very much.
Thank you for having us. Bye.